Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. How are you all? Good evening all of you. And guys, in today's session we are going to complete plant physiology. All the three chapters, photosynthesis, respiration and plant growth and development will be completed in today's session. So quickly buckle up, get your notes. We are going to make quick notes along with me, and of course you will get the PDFs. I have brought a lot of lot of flowcharts for you so that things get easier for us to learn and revise. Even those of you who are finding it difficult to learn things, even at this hour, can learn from these flowcharts. Good evening, all of you. Good evening. Quickly share the session with all of you, all your friends, and give me a thumbs up that you're ready with your notebook or your NCERT or the notes that you've already prepared. Good evening. Quickly give me a thumbs up. Good evening. I am fine, and thank you so much for asking. I'm feeling better today. Good evening. Glittering twilights with hue, drizzle, ma'am. Hope you're doing great. Yes. Wow. How poetic is that? Frog, I was not feeling well yesterday or at all. I don't know whether you realized it in the class or not, but I was not feeling well. So I could not. I'll do that. I'll do that today positively. Good evening, everyone. Session duration, it will be, of course, at least 2.5 hours. It should take. It may go up to 8, but not beyond 8. I know there's a different session at 8, so I'll try to wind up before that or max at 8. Okay, now let's begin. Okay, let's start with the first chapter. But okay, I am going to start in this. I'm going to do plant physiology in this order. First of all, plant growth, then photosynthesis, and then respiration. So respiration is a very uh, tricky chapter where you do get questions in the form of numericals also. So you need to have a very clear understanding of the topic. So stay with me, and there are few numericals that you get from photosynthesis also. So we are going to discuss all those tricks, we are going to discuss all those topics, the flow charts of respiration and everything in today's session. Good evening. All right, shall we start? Please let me know. Shall we start? All of you give me a thumbs up that you're all ready, and we can begin with the session. Previous question paper, yes, we will be discussing all of it. Give me a thumbs up. All right, Charu, you're welcome from Himachal. I am from Punjab. I'm basically from Punjab. Yes. All of you are set? Shall we start? Where are the green hearts? I'm not getting green hearts. Enough green hearts. All right. Let's begin with the session. So the first flowchart is right in front of you. All right, guys. So it is about plant growth and development. First of all, we are going to talk about, see, there are two terms. One is growth and second is development, right? First of all, let's talk about growth. So what does NCERT say? That of course it is a characteristic of living being. This is what we have read in chapter number 1. And if you remember, we have discussed that growth and reproduction are the non-defining features. They are not the defining features. Why? Because even non-living organisms can show growth. The difference being that living beings they show intrinsic growth, but non-living beings show extrinsic growth. But we do know it is a characteristic of living being and it is non-defining, right? Secondly, growth is an irreversible change. Once you are grown up, you cannot go back to the initial stage. Are these points clear? All right. Huh. All right, moving again, then we have high metabolic. So growth demands a high metabolic activity, right? Why? Because there's a lot of breakdown and build up processes going on. So it involves both catabolism as well as anabolism. Now in plants, guys, growth is indeterminate. Why is it indeterminate? Because it's not fixed. Like in case of animals who have a fixed growth, our growth is not fixed. It is indeterminate because of the presence of meristem. And we know there are meristems present at different places. Now in case of dicots, if you remember, there is secondary growth also possible because of the secondary meristem, and we call it open growth. We have read anatomy. Yes or no? All of you are clear with this? All of you are clear with this? All right. Then, guys, growth is increase in size. So there are actually different parameters of growth, and the growth can be measured. We know that growth can be measured, and there are different parameters in which this growth can be measured. So, for example, growth is increase in size. It is growth increase in weight, increase in length, or it growth can be regarded as increase in the number of cells or surface area. So all these different parameters indicate growth. Are these points clear to all? All right. Moving on to the parameters again. Now let's talk about these different parameters in detail. Now we say growth is measurable and there are certain parameters. For example, fresh weight, dry weight, number of cells. It can be measured in terms of volume, then length, and then surface area. Please let me know that these points are super easy. Super easy. So talking about the fresh weight, see, fresh weight, instead of using the fresh weight, we prefer using dry weight as a parameter. Why? Because in dry weight, the water has already been removed, right? So on drying, all we have is the solid matter which is present. It is no water weight. Water is removed. So that is why dry weight is always considered more accurate than the fresh weight. So dry weight is a better and a more authentic parameter as compared to the fresh weight. Is this clear? So whenever we are measuring the weight of the body or an organ, we prefer dry weight. Then coming on to the number of cells. See, for this, NCERT is given a very important example. See, one maize cell will increase up to 17,500 cells in an hour. So imagine the rate of growth for maize cell. So in maize cell, we measure growth in terms of number of cells, how much number of cells have increased. Then in case of melon, in case of watermelon, the growth is indicated by increase in the size of the cell. And in watermelon, the size can grow up to, it can increase up to 3,50,000 times. So you have to remember how growth is indicated in maize cell and how growth can be indicated in the watermelon, sorry. Then increase in length, this is seen in case of pollen tube. So whenever we are trying to see if the pollen tube has grown or not, what do we measure? We measure the length of the pollen tube. So in pollen tube shows growth in terms of increase in length. And then for surface area is used as a parameter for studying the growth in the leaf. So we always see how much broad it has become. So in that case, in leaf, we use the surface area. Please tell me that all these points are clear to you. Please tell me all these points are clear to you. Yes? All right. Moving ahead to the growth rates. So see, I have already discussed complete NCERT with you, class 11 as well as class 12. So make sure that you keep revising that NCERT. So we completed NCERTs in hardly 4.5 hours. So now if you increase the speed, you can revise a complete NCERT multiple times before going for your final NEET exam. Yes. So keep doing that along with these sessions again and again. Right now, see, there are two types of growth rates seen. Two types of growth rates are seen. So if the increase is very gradual, like 1 to 2, 2 becomes 3, 3 to 4 and 5. So when there is gradual increase, this is called arithmetic growth. And when it doubles up at every level, this is known as geometric growth. Now NCERT has said, see, if there is a parent cell, and when the parent cell divides by mitosis, of course, there are two daughter cells formed. One is A, another is B. So if only one divides and the other goes for differentiation, other doesn't divide, because if you have to differentiate, you cannot divide, right? So A undergoes differentiation but does not divide. So basically what is happening, only one divides and other differentiates. So at every division, at every level, only one is added up. So 1 becomes 2, 2 becomes 3, 3 becomes 4, and so on. So this kind of growth is known as arithmetic growth. And for that, what kind of curve do we get? We get a straight line. We get a straight line. So this linear, this graph, sorry, arithmetic growth always gives us a linear curve. So it is a linear graph. What is the shape of the graph here? Linear. Is this clear? And what is the equation for this? The equation is LT is equal to L naught plus RT. L naught is the original length. Are these points clear? Please tell me, are these points clear? And then guys, we are going to talk about the geometric growth. So for geometric growth, this is the formula. So LT is equal to L naught ERT. You can take any parameter. If I want to measure geometric growth in terms of weight, just the alphabet L will change into W. So WT is equal to W naught ERT. Are you clear with everyone? Everything? Yes, please. Everyone, are you clear? So basically, when does geometric growth takes? Geometric growth is going to occur when the parent cell divides mitotically. Parent cell divides mitotically, and both the daughter cells retain the ability to divide. Both daughter cells retain the ability to divide. When both will keep on dividing, see, A will also divide further, B will also divide further. So with every step, the number of cells increasing are actually doubling up. The number of cells which are getting added is double. Yes, please. Please let me know if these points are clear to you all. Please let me know these points are clear to you all. Guys, why are you spamming? Please concentrate on your work now. How does it matter if anybody says anything in the chat? Useless. Okay, coming back here. So embryos. This is very, very important. I've already discussed this in NCERT reading also. Embryo, what kind of growth is shown by embryos? Embryo shows both arithmetic as well as geometric growth. So at different stages of embryonic development, different types of growths are visible. Please let me know that these points are very, very clear. Please let me know these points are clear. Moving on to now different types of growth rates. Quantitative comparison. Now let's make a quantitative comparison of growth rates. So if we go for the quantitative comparison, guys, we have absolute growth rate and then we have relative growth rate. Now for this, let's take an example. So we have this one leaf, A leaf, and we have a B leaf, right? So this A leaf is initially 5 centimeters square. The surface area is 5 centimeters square, while this B leaf has the initial surface area as 50 centimeters square. Now this A leaf becomes 10 centimeters square on growth, and this B leaf becomes only 55 centimeters square on growth. Are you getting that? It becomes 55 centimeter. Now, what is absolute growth rate? You have to see the formula. Absolute growth rate is equal to final minus initial. That's it. So, what is the final here? 10. What is the initial? 5. So, what is the absolute growth rate in case of A? The absolute growth is 10 minus 5, which is 5 centimeters square. And what is the absolute growth rate in case of B leaf? That is 55 minus 5, 50. That is 5 centimeter again. So in both the cases, the absolute growth rate is same for leaf A as well as leaf B. Are these points clear? But now if I talk about relative growth rate. Now when we are talking about relative growth rate, there is a common parameter. So for the common parameter can be initial growth, initial surface area or initial length. So there has to be a common parameter. See, common parameter for studying for studying the relative growth rate. Are these points clear? So relative growth rate is equal to final minus initial upon initial. Is that clear? And into 100. So final minus initial is same 5 for both the cases, but here for A leaf 5 upon 5 into 100, it would be what would that be? 100% increase. But for here, if I see 5 upon 50 into 100, that would make it just 10%. So absolute growth rate is same, but relative growth rate is different for both the leaves. Do you understand the difference? And have you seen the formula here? So guys, the formula are very, very important. The value might be different in exam, but the formula remains the same. Please give me a thumbs up that all these points are clear to you all. Give me a thumbs up. Yes, guys. Are you clear? Are you writing down all these points? Perfect. Moving on to the conditions for growth and development. So see, there are two types of conditions. Of course, there are external conditions called extrinsic and internal conditions which regulate growth are called intrinsic. Now, the extrinsic conditions are light, temperature, water, oxygen, and nutrients. And we know that some nutrients are needed in large amount. We call them macronutrients, which are needed in small amount. They are known as micronutrients. Is this clear? Then coming on to the intrinsic factors, the internal factors. One is the genetics. So the genes regulated. And second are the hormones which are produced inside or by the plant. These are known as phytohormones or plant growth regulators. Genetics is an intracellular condition or a factor, while for phytohormones are intercellular. Now please see the difference. Intracellular means genes are internal matter of every cell, while phytohormones can be secreted from one part and will act on the other part. So they work between the cells. Are you getting that? Please let me know. Please let. All right. So are these points clear to everyone? Moving on to the next factor. Now, next terms which are given in your NCERT in this chapter is differentiation, de-differentiation, and redifferentiation. So we already understand what is differentiation. When the cell becomes specific. So from meristem, the cell becomes specific to perform a specific function. We call it maturation of the meristematic tissue, right? So it undergoes major structural changes in the cell wall as well as in the protoplasm as per the need. So what it is specializing in, for example, tracheary elements. For example, tracheary elements of vessels, they lose their protoplasm. So we know that tracheids are tracheids are dead in nature, right? So they die when they are maturing. They become dead. So why do they become dead? Because they lose their protoplasm. Then lignification takes place, and they become strong and elastic. So see, these are all changes which a cell is undergoing for the sake of maturation, for performing a specific function. Then, in case of need, the differentiated cell can become undifferentiated again and become meristematic. So basically, a differentiated big cell becomes de-differentiated and becomes so permanent cell. A permanent cell becomes meristematic again. This is known as de-differentiation, right? So when a meristematic cell becomes permanent, this is differentiation, and in times of need, when permanent becomes meristematic again, this is known as de-differentiation. So there are some examples of de-differentiated tissues. We call it cork cambium, interfascicular cambium. Now remember, a permanent tissue when divides a permanent tissue when de-differentiates to become meristematic, such meristematic tissue is always known as secondary tissue. It is always known as secondary tissue. Are these points clear? Then, once the need is fulfilled, these meristematic cells can again become differentiated. So when they again become differentiated, this is known as redifferentiation. This is known as redifferentiation. So secondary meristem will again lose the ability to divide to perform a specific function. This will be known as dedifferentiation. And now the permanent formed out after, see here. So this secondary meristem becomes permanent again, right? Now the permanent formed from secondary meristem will be known as secondary permanent tissue, or we call this process as secondary growth. Do you remember that? This is known as secondary growth. Please tell me all these points are clear to everyone. So they lose the ability. So we have examples like cork, bark, and new vascular bundles which are produced to increase the girth. They all come under the example of redifferentiation. Please let me know all these points are clear to you all. So guys, as you know, we have already started with the mock test series. Yes. So one has already happened on Sunday, that is 19th, and from this Sunday, that is 26th, the mock tests will be regular. Mock tests would be regular, and see, even NTA is insisting to all the students that you should take a mock test before coming for the real NEET exam. This is so important. Even NTA understands that, because it is very pressurizing to take that test in the first time. So why not go through that whole process before and get your mind ready? And also it will give you a glimpse into your preparation, and you still have time. Imagine one week to work on your shortcomings. Yes. So do take that take the this free mock test series for NEET UG 2026, absolutely free on our app. Absolutely free. Is that clear? Okay, moving ahead to the second part. See, the chapter is plant growth and development. Now, coming on to the second term, what is development? Development means it is a sum total of all the changes that occur. It is not just growth, it is growth plus other things, right? So, development depends on the stage of the life and the environment. So, see, a plant starts with seed, right? So seed germinates, seed grows, it grows into a proper plant. Then that plant and starts undergoing aging. As soon as a plant comes out, as soon as we are born, we start aging, right? So senescence is something that goes along the entire life. Similarly, germination of seed, then followed by senescence, also known as aging, and finally death. So see, there are two important aspects of development. One is growth, and second is getting mature to perform a specific function, which is known as differentiation. Are these points clear to everyone? Please let me know. Are these points clear to everyone? Then, what is plasticity, guys? What is plasticity? So, plasticity is the different response of the plant towards different phases of life and environment. So, basically, if the phase changes, now the structure will also change, or the environment changes, the structure and the response will also change. Now, one such example of plasticity is when leaves are present, so the leaves show different structures. So when different type of leaves are present in the same plant, this is known as heterophily. It is one example of plasticity. Is that clear? For example, see, so plasticity or heterophily can be seen under two conditions: different phases of life and in different environments. Now there are different examples. See, cotton, coriander, and larkspur. So these three plants will produce different types of leaf during different phases of their life. Is it clear? For example, these cotton, coriander, and larkspur, they'll have this less dissected kind of leaf in their early phase, in their juvenile phase. But once the plant is adult, it will have more dissected leaf. Are you getting? So this changes with the phase of life. This is also heterophily. But in case of buttercup, we see different leaves depending upon the habitat that it is in. If the buttercup is placed in a terrestrial habitat, it is on land, it will be less dissected. Okay, but if the same buttercup is now grown in aquatic habitat, it will be highly dissected. Are these points clear? Are these points clear to all? Please let me know. So this is known as heterophily. Then moving on to plant growth regulators. See, plant growth regulators, also known as phytohormones. So we are going to study 5 different plant growth regulators: auxins, cytokinins, gibberellins, ethylene, and abscisic acid. Now, out of these five, auxin, cytokinins, and gibberellic acids, these three are the promoters, while ethylene and ABA, that is abscisic acid, these are the inhibitors. Are these points clear? Please let me know, are these points clear? Okay. Now, first, let's see the source. How is auxin produced? What are the sources of these plant growth regulators? So, indole compounds. Auxins are the indole compounds, while cytokinins are formed from or produced from adenine derivatives. Gibberellins come from terpenes. Ethylene is a gaseous one, and abscisic acid is a derivative of carotenoids. It's a derivative of carotenoids. Clear? Then, second thing, see, this one flowchart has lots of information. So, please use them well. Clear? Now, talking about the discovery. The discovery of auxins was made by Charles Darwin and his son Francis Darwin, and they discovered this auxin from the polyptyles of canary glass. Okay, so they discovered it from the canary glass, but auxins were first isolated. See, isolation was done by F.W. Wendt. Isolation was done by F.W. Wendt from the polyptyles of oat or avena. Is this clear? Please let me know that all these points are clear to everyone. And while Charles Darwin and his son were discovering it, this is when they discovered the phenomena of phototropism being. So they explained this phenomena of phototropism. They discovered the reason behind the phototropism. So that is how auxin got discovered. So everybody knew that plants grow towards light, and the stem bends towards the light, but nobody actually gave us the reason. So the reason behind phototropism was identified or discovered by Charles Darwin and his son. Then coming on to cytokinins, adenine derivatives, also N6-furfuryl aminopurines. Is this clear? Adenine is a purine, so it is N6-furfuryl aminopurine from which cytokinins are derived. So cytokinin was discovered by S.F. Skoog from the tobacco plant. Then when he said, see, not exactly from the tobacco plant. So that is when Skoog said, if you put the mixture of of coconut milk or the vascular bundle, so when you put the coconut milk in tobacco stem, it grows faster, right? In the callus of tobacco stem, it grows faster. So that's how it got discovered. Then coming on to gibberellins, terpene derivatives discovered by Kurosawa, a Japanese one, of course, and he discovered it from a fungus Gibberella fujikuri. How did he discover it? He discovered it when he saw that this fungus led to elongation of the rice seed. So foolish seedling disease of rice. Is this clear? We call this disease as Bakkane's disease. So it was discovered that this fungus is causing that foolish elongation of the wheat of the rice seed. So Bakkane's disease, and that's how it got discovered. So basically, this fungus Gibberella is producing a certain chemical leading to foolish seedling disease. Is that clear? Then moving on to ethylene, which is a gaseous compound and discovered by H.H. Cousins, and they discovered it in ripened oranges. So they saw that if ripened oranges are kept near unripened banana, so the ripening process of banana gets enhanced. And finally, abscisic acid, it is a carotenoid derivative discovered by three scientists independently, and they named them differently also. One called it Dorman, second one called it abscission 2, and the other one called it inhibitor B. But later on, all the three different compounds were not found different. They were chemically just the same compound, that is ABA, abscisic acid. Are these points clear to everyone? Now, moving on to the discovery of auxins, I have already explained this, and this was done by Darwin and his sons by chance. And this discovery happened by chance. He was not actually looking for anything. It happened by chance. So such discovery is known as serendipity. And we see that the plant, so the tip of the plant grows towards the light. So what did he do? He made a unilateral illumination. So when there was unilateral illumination of light, the tip of the canary grass started growing towards the light. So basically what happens, see, what did he conclude? So auxin is produced at the on the side opposite to the light. Remember that. What happens? So what do auxins really do? Auxins are produced. So this line is important. Auxins are produced on the side, on the opposite side of light. Opposite side of light. And see, auxins produced on the opposite side, and this causes cell growth and elongation on the opposite side. So the cells on the opposite side of light, they grow, they elongate, and this results in bending of the stem towards the light. Are these points clear to everyone? Are these points clear to everyone? Please let me know. There is no lag from my end. All right. Then discovery of gibberellins. Already told you Kurosawa did that from foolish seedling disease. He saw that if the rice grain is infected by this fungus, Gibberella fugiculae, then it leads to the foolishly elongation of the seed, right? And from this fungus, from the sterile filtrate of the fungus, they were Kurosawa was able to find out this chemical. He was able to extract gibberellic acid. He was able to extract gibberellic acid. Are these points clear? Then discovery of ABA, already told you by 3 independent researchers in mid-90s, but later on they were found to be same chemical. Already told you H.H. Cousins made the discovery of ethylene, that if ripened oranges are kept near the unripened banana, then the process of ripening is hastened, it is increased. Then discovery of cytokinins. Now, how was discovery of cytokinins made? So basically, a callus or undifferentiated mass of cells was taken. This undifferentiated mass is known as callus. So when callus was mixed with, so we added auxins to the callus, but along with auxins, when the callus was given extract of vascular tissue or the yeast extract or the coconut milk. So along with auxin, if we added one of these three things, vascular tissue, yeast extract or coconut milk, it was seen that differentiation of root or the stem happened quickly. Is it clear? Later on, it was Skoog who found out that basically these three things, which were added in addition to auxin, they were sources of cytokinesis and cytokinins, and they enhanced the rate of cell division. Is this clear? Is this point clear? So this callus thing is used in tissue culture. So remember, while doing tissue culture, if you add, please see this, if you add more of auxin as compared to cytokinins, then rooting will begin first. But if the level of, or if the sorry, if if the ratio of cytokinins is more as compared to auxin, then shooting will begin first. So remember Ka. See this. So this ratio is important. So if auxin to cytokinin ratio is high, then it leads to rooting, and if cytokinins are more, then it leads to shooting. So can you remember ARCS? So auxins more, then rooting, cytokinins more, then shooting. Can everyone remember that? So this is the discovery of cytokinins. Moving on to now their physiological effects, and now these flowcharts will be very, very helpful to you in remembering and revising the things. So first of all, see, there are two types of auxins. There are two types of auxins. Yes, there are two types of auxins. So one is natural auxin, which are naturally produced by the plant body, which is IBA and IAA. Indole butyric acid and indole acetic acid are the natural auxins, while there are some chemicals also which act like auxins. They are known as synthetic auxins, that is naphthalene acetic acid, Na, or 2,4,5-T, 2,4,5-trichlorophenoxyacetic acid, or 2,4-D, which is 2,4-dichlorophenoxyacetic acid. Now, these auxins are always produced at the tip of the shoot and tip of the root. Is it clear? Now, what are the functions? First of all, auxins are known to initiate rooting in the stem cutting. So when we are performing the vegetative propagation and on the stem cutting, if we spray some auxins, then rooting will become easier. Then it promotes the flowering in pineapple. Remember, then prevents early drop of fruits and leaves, Induces parthenocarpy, especially in tomato we have seen it. And when you induce parthenocarpy, we are able to produce the seedless varieties of food. So fruit without fertilization will form by the help of auxins. And one such application is done in tomato. Then there is apical dominance. So what does apical dominance means? So till this tip keeps on growing, till this meristematic thing is there and the tip keeps on growing, the lateral branching doesn't happen. So the apex dominates over the lateral buds. This is called apical dominance. This is because of auxins. Then xylem differentiation and cell division in callus, and also 2,4-D particularly, it acts as 2,4-D acts as a herbicide or a weedicide, and only for the dicot weeds. Only for the dicot weed. So 2,4-D will prepare the weed-free lawn, but that weed has to be a dicot in nature. Are these points clear to everyone? Please let me know. So guys, with this, auxins are done. So already told you what is apical dominance. So till that shoot tip keeps on growing, it will dominate over the lateral branching. So if you cut the tip, the cutting of the tip is known as decapacitation. Cutting of the tip is known as decapacitation. So when decapacitation is done, apex will not dominate anymore, and so now there will be increase in the girth. So apex, till the apex will dominate, the girth will not increase. But after the decapacitation is done, now tip stops growing and rather it starts profusely branching. So the lateral development will take place. The lateral buds will now grow. This leads to increase in the width. Now this method is used for hedge making and tea plantation. Auxins have their role in hedge making and tea plantation. Please let me know. So how can apical dominance be suppressed? How can apical dominance be suppressed? One method is decapacitation, just cut the tip of the shoot. Also, we'll see that instead of decapacitation, if we use cytokinins, then cytokinins are antagonistic to auxins in this manner, and cytokinins will help us in overcoming the apical dominance. Now, these are very, very important lines. So I hope these flowcharts are helpful. Will you be using them to revise? Let me know quickly. Is that clear? So the PDF will be uploaded. Use these PDFs, use these flowcharts to quickly revise. All right, guys. Now moving on to the second, that is gibberellin. So what do gibberellin do? Let's first know more about gibberellins. These are acidic in nature. They are called gibberellic acid because of their acidic nature. More than 100 types of gibberellins are known, and we name them as GA1, GA2, GA3. Now, GA3 was the first form of gibberellin to be discovered and most widely studied. So the first gibberellic acid which was discovered was actually GA3, but isolated later on. Is that clear? Now, what are the functions? First of all, see, gibberellins are known for elongating something, right? They increase the length of the plant axis, for example, length of the grape stalk. So this grape stalk becomes this elongated, right? Then it is used for changing the shape of the fruit. So improvement of fruit shape. For example, if this apple is like this, it will become more like this. Is that clear? So gibberellins are known for elongation. Then internodal elongation also. Now internodal elongation is seen particularly in stems like sugarcane. So let's say if we have this sugarcane, this is a sugarcane. So when the internodal elongation would occur because of gibberellins, what is going to take place? Increase in the length, yes or no? Now gibberellins can increase the yield up to 20 tons per acre. So if you use gibberellins along with the sugar cultivation, your yield can go up to 20 tons per acre. Is that clear? Then enhances the malting process, that is during the alcohol production brewing industry. Hastens the maturity period in conifers, so early seeds will be formed. It delays aging. As we know, auxins, gibberellins and cytokinins are promoters, growth promoters. So they delay aging, they delay senescence of fruits, thereby they increase the shelf life of the fruit. So the fruit is not going to ripen so easily. So the market period of a fruit is increased. And then talking about the bolting process. What is bolting process? Internodal elongation before flowering is called bolting process. Are these points clear to all? Please let me know. Please let me know. Are these points clear to all? Yes or no? And the examples of bolting process are beetroot, cabbage, and rosette plants. Are these points clear to everyone? Guys, what are you talking about? Grumpy ship and all. Please concentrate. Please concentrate. Now see, talking about the third, which is cytokinins. So cyto means cell and kinin means divisions. The word comes from cytokinesis, right? So what could the function of cytokinins be? These are going to enhance cell division. They are going to increase the rate of cell division. Cell is going to divide or multiply faster. See, it was first discovered in the form of kinetin. So cytokinins were first discovered in the form of kinetin. So although kinetin does not naturally occur in the plants, so extracted kinetin was actually, this form of cytokinin was extracted from an autoclaved herring sperm DNA. What is herring? Herring is a fish. So kinetin was the first form of cytokinin to be discovered, but not from the plants because kinetin doesn't occur in plants. Are these points clear? In plants, rather, plants have cytokinins in the form of zeatin. Is it clear? So, sorry. Cytokinins in plants occur in the form of zeatin. Why do we call it zeatin? Because it was first discovered in zea maize. Zea maize is maize. So naturally occurring cytokinin is plant and was isolated from corn kernels and coconut milk. That is why, if you remember, Skoog, when he added this coconut milk along with the auxin to the callus, it resulted in the differentiation. Is this point clear to everyone? So the natural form of cytokinin in plants is zeatin. So what else? What does it do? It promotes cell division in root and shoot tip. Already understandable. Development of shoot buds and young fruits, formation of new leaves and chloroplast. Then it counteracts apical dominance. Just explained you. If you want the plant should stop growing vertically upwards and rather start increasing the girth. That means the lateral branching should happen. Then either you decapacitate it, cut it from the top, or you spray cytokinins. So it helps in counteracting the apical dominance and leads to the lateral growth. Then again, being a promoter, it delays the leaf senescence by mobilization of nutrients. Mobilization of nutrients means the nutrients from the old leaf will be transported or mobilized to the younger parts of the plant. And then the development of adventitious buds, which will lead to lateral branching. This is all done by cytokinins. Please let me know that these points are clear. Then coming on to the fourth, that is ABA or abscisic acid. It regulates abscission and dormancy and inhibits the plant growth and metabolism. So first, it inhibits the seed germination, known as the stress hormone, so stimulates the closure of stomata during the water stress. Important role in seed development, maturation and dormancy. Helps to withstand the unfavorable conditions. So that's how it is also known as the stress hormone. So it prevents the dehydration. Is that clear? And it is antagonistic to gibberellic acid. So far you are known to two sets of antagonistic hormones. One is auxin and cytokinin. Auxin causes apical dominance, cytokinins are against it. So they help in overcoming the apical dominance. Second, ABA and gibberellic acid. So these are the two sets of antagonistic hormones. And last not the least, we come on to this fifth phytohormone, which is ethylene, produced by the tissues undergoing senescence and ripening. This ethylene is gaseous, that is volatile, but the form of ethylene that we use for plants, for ripening the plants, is ethaphon. It is ethaphon. So what do we do basically? Ethaphon comes in the form of a tablet. It is dissolved in water and then this water is sprayed all over the plants. Is this clear? So see here. So used in the ecosperm compound and this water which contains this ethaphon tablet, this is going to release ethylene slowly on getting dissolved in water. Are these points clear to all? Please let me know that these points are super clear. And now, guys, moving on, it promotes the root growth and the root hair formation. Fruit set in pineapple causes flowering in mango. Then particularly for ethaphon, it is written, it promotes the female flowers in cucumber, accelerates abscission in flowers, then thinning of cotton. This is important. Thinning of cotton, cherry and walnut, hasten the fruit ripening. See, which hormone is responsible for hastening the seed maturation? That is gibberellin. But hastening fruit ripening is because of ethylene. Is that clear? Then petiole and internodal elongation in deep water rice plants. So the rice plants are quickly going to grow taller if we use ethylene. So this will help the rice plant to grow out of the water. Is that clear? Since we know that there is water going to stand in the paddy fields, so this ethylene is going to help the plant to grow faster and come outside the water. Then horizontal growth of the seedling, swelling of the embryonal axis and the formation of apical hook in the diploid seedling. Also, it leads to respiratory climactic. What is respiratory climactic? So it enhances the rate of respiration in fruits during ripening. So when when the fruit is ripening, the metabolism is very high. It is energy demanding process, right? Ripening is an energy demanding process. So this enhanced respiration rate during the ripening process is known as respiratory climactic. And which hormone helps in reaching to that? It is ethylene. Clear? Then last not the least, it breaks the seed and bud dormancy, and seed germination will occur in peanut sprouting of potato. Please let me know that these points are clear to you all. Then coming on to the action of hormones, the hormones can be very individualistic. So they have their own effect, right? There are certain hormones which work better with the other hormone. For example, we saw that auxin and cytokinins both help in callus development. Yes, so that is when they are acting synergistically. While complementary, action of the hormones can be complementary also. They add to each other's work. And antagonistic, like we saw auxins and cytokinins, gibberellic acid and APA. So these are the two antagonistic hormones. Are these points clear to everyone? Please let me know all these points are super clear to all. And with this, guys, we complete our first chapter of plant physiology. And now moving on to photosynthesis. Yes, please. Then, not just only theory questions. Yashwant, this is unit-wise revision. Going on. First, let's revise all these questions. Sorry, the chapters. I am discussing the whole chapter. Not even missing on one line from NCERT. Okay, so now questions will be practiced very soon. I'll be soon starting to take some classes, question-based classes on Unacademy app. In special classes. They'll be free and everybody can join. Now, starting with the second chapter of plant physiology, which is photosynthesis. So we understand the process. Guys, I can actually claim those of you who have not been able to understand certain processes during the entire 2 years, they can understand the process through these flowcharts in these sessions now. Even at the last moment, you can do that. And how much you have studied, how much you have retained, you should start checking on that. Even NTA is telling the students through their, they're releasing these notifications that students are advised to take the mock test before coming for the real exam. So now the link is given in the chat box also. You can see that the message pinned in the chat box. Click on it and register for that. Register for the free mock test series from Sunday onwards, that is 26th. They will be happening every day 2 to 5 p.m. So make sure that you take every test and nobody misses on it. Is that clear? This is very important. And before 26 also, you should also be practicing the mock test on your own. You can ask your doubts rather than talking just random things in the chat. You should be discussing things with me. And as you know, I always see your chat. I know what's going on here. Notes will be available on the Telegram channel. But rather than finding my notes, you should be focusing on making your own notes. See, what you write on your own will be just inscribed so strongly in your memory. You'll exactly know, okay, I wrote this in the class. That's how it happens. Now, coming back here, and I don't want to see anyone making any useless comments here. Please focus, guys. Please focus. Now, starting with photosynthesis. So, site of photosynthesis is leaf, right? And majorly, we know if we talk on the cellular level, it is the chloroplast. But let's go step by step. So, leaf is the main site of photosynthesis, and we have studied anatomy already. It is the palisade layer of leaf which has maximum chloroplast, around 20 to 50 chloroplasts per cell. Why? Because this is one of its adaptation. We have seen what happens during differentiation. There is change in the protoplasm. So the palisade mesophyll has developed a large number of chloroplasts because it is meant for the sake of photosynthesis. Is that clear? Now the alignment of chloroplast will be also different as per the intensity of the light. Is this clear? Now see, if the light intensity is low, the chloroplast lies like this. So see, these are NCERT lines. Chloroplasts align themselves along the wall of mesophyll such that they get optimum light quantity of incident light. And when do you think the chloroplast will be aligned with the flat surfaces parallel to the wall? So they are parallel to the wall when the light intensity is low, and they are perpendicular. They are perpendicular to the incident light when the light intensity is high. Why? Because you must know that too much light, too much intensity of light can also destroy chlorophyll, can also destroy chlorophyll. Is this clear? Uh-oh, just a second. Yes, guys. Let's first understand the main side of photosynthesis, which is chloroplast. See, it is double membrane structure. It has a double envelope, right? It has its own DNA. It has its own 70S ribosomes. These are of prokaryotic nature because it is semi-autonomous. This chloroplast has the ability to make its own proteins, some of the own proteins. So it is semi-autonomous. Okay, this is something we have already discussed yesterday. I hope all of you have seen unit of cell and revised it well. In case anybody has not, please do that as soon as possible. Now, coming on to the other structures which are of more relevance for this chapter. So, there are some flattened sac-like structures known as thylakoid structures, All these flat structures are thylakoid. So, there are thylakoids stacked one upon the other. So, the thylakoids, when they are stacked one upon the other, they form this structure called grana. So individual structure is thylakoid and the stack is known as grana. Now all these grana are interconnected by the help of fret membranes or stromal lamella, and the surrounding area has this matrix, and this is known as stroma. Now we already know, since we are not new to anything here, stroma is the location or the site for dark reaction, while this thylakoid and grana, these and even stromal lamella, these are the sites for light reaction. Are these points clear to all? Please let me know. Now the pigments which are going to trap light, those pigments are found in the thylakoid membrane like this. So there's going to be a whole pigment system that we will study. So it is in the walls of thylakoid membrane that the pigment molecules are present. Clear to all? Now, first of all, let's understand the pigments. Before going into the details how they work, let's understand which all pigments are present and how they are present. So what are pigments? So first of all, pigments are always the light absorbing chemicals. So anything which can absorb light, right? We call it a pigment. Is this clear? Then, here. So light absorbing chemicals are called pigments. So there are different types of pigments. For example, we have chlorophyll, we have carotenoids, right? So chlorophylls are further of 5 types. We have chlorophyll A, B, C, D, E. We don't have to go there. Just remember chlorophyll A. Chlorophyll A is found in all the photosynthetic organisms, be it cyanobacteria, be it euglenoids, and now be it our green plants. Clear? So that is why it is called a universal chlorophyll. Is that clear? Now, why do we call it? This chlorophyll A also forms a reaction center. Okay, this chlorophyll A is called the reaction center because it absorbs light most effectively, and that is why it is most abundant also. Clear? Now, this becomes an assertion reason. Please watch. Rest of the pigments will be present in different plants in different concentration, but A is present in the most abundance. This is most abundant chlorophyll. Please tell me that these points are clear. Now, apart from chlorophyll A, rest of the chlorophylls and your carotenoids, carotenes as well as xanthophylls, they constitute the accessory pigments. Now, these are known as the accessory pigments. Is this clear? Now, what do we understand by accessory pigments? Why are they needed? These accessory pigments will absorb extra wavelengths of light, and ultimately they are going to absorb that light and transfer it to chlorophyll A only. Then second, they are present around the chlorophyll A. See, this is chlorophyll A, and then surrounding chlorophyll A are present the accessory pigments. Is that clear? These are the accessory pigments. That means other chlorophylls and then carotenoids and xanthophylls. These are the accessory pigments. Are these clear? So they are surrounding the chlorophyll A, and they will protect chlorophyll A from getting destroyed by high intensity of light. So they protect from photo oxidation. Are these points clear to all? Then third, by absorbing extra wavelength, they are basically going to increase the efficiency of chlorophyll A and increase the productivity of photosynthesis as well. Please let me know that all these points are clear to you all. Now see, this is how the pigments are arranged. So this is the thylakoid membrane. Now thylakoid is basically a single membrane structure. Thylakoid is a single membrane structure. But we can definitely demarcate the outer side of thylakoid. So this becomes the outer side, this becomes the inner side, but the membrane is only single. Please understand. So even if it is a single membrane, this is the outer side, this is the inner side. Can we say that? I'm not calling it outer membrane, I'm calling it outer side of the thylakoid membrane. Now, why do we need to see to these details? Because the different things will be present at different locations, and you must know the location of everything. Now, see here, there are questions based on that. So, let's come back to this. So, in the center, guys, is present chlorophyll A. Is present chlorophyll A, and surrounding it, see, these are accessory pigments as well as some proteins. Now, accessory pigments plus proteins, they constitute the antenna molecules, which is also known as light harvesting complex. Also known as light harvesting complex. Is this clear? The chlorophyll A present in the center becomes your reaction center. So now, reaction center, that is one molecule of chlorophyll A surrounded by light harvesting complex or antenna molecules, in total forms a photosystem. In total forms a photosystem. Please tell me that these things are clear. What am I saying? It is a photosystem. So photosystem has two things. One is reaction center made up of chlorophyll A, single molecule of chlorophyll A, and then there are light harvesting complex or antenna molecules. And what is this made up of? Accessory pigments plus proteins. Please tell me that these points are clear to you all. So this constitutes a photosystem. Is that clear? Now we have two types of photosystems. One is photosystem 2 and then we have photosystem 1. Now remember, this 1 and 2 is in the order of their discovery. So photosystem 1 was first discovered and photosystem 2 was discovered later on. Are these points clear? Now what is the difference? The difference is in the chlorophyll A. This chlorophyll A can be of two types. One type of chlorophyll A can absorb light up to a maximum 680 nanometer. So is that clear? And then there is another type of chlorophyll A which can absorb light up to or maximum up to 700 nanometer. Are these points clear? Are these points clear? Right? So the photosystem 2 has chlorophyll A with maximum 680 nanometer absorbing capacity, while the photosystem 1 has a chlorophyll A with maxima of 700 nanometer. So we call this chlorophyll A as P700 and we call this as P680. Are you getting that? So what does this number written in the subscript of P mentions? What does it indicate? So 680 means maximum light intensity that it can absorb is up to 680 nanometer, and P700 has a higher ability. So maximum light absorbed by this one is up to 700 nanometer. Please tell me that these points are clear to you all. Please tell me these points are clear to you all. Right? Okay. Now, when you have understood where all these characters are located, please see, this is very, very important. And by the way, the reaction center is towards the inner side of the thylakoid membrane. Okay. This chlorophyll A center, reaction center is towards the inner side of the thylakoid membrane. Are these points clear? Okay. Now, this is chlorophyll A, universal pigment found in cyanobacteria, euglenoids, and all plants. And there are two types already discussed. So we can actually, so how do we know that there are different types of chlorophylls present? So we have a technique known as paper chromatography to differentiate, to separate, okay, and study and see their presence. So by paper chromatography, we separated these pigments A, B, carotene, and xanthophylls, right? And when we saw that different colors, we saw that chlorophyll A is blue-green in color, B is yellowish-green, carotene is orange-yellow, and xanthophylls are lemon-yellow in color. And this is a very important PYQ. You've already gotten this question in Match the Hallway. Is it clear? Second point. Now see, you get these action spectrum and absorption spectrum also. What do we understand by this? Absorption spectrum basically tells us about which which wavelength of light is maximum absorbed by these three pigments. So we are going to talk about chlorophyll A, chlorophyll B, and carotenoids. Okay. So when you study the graph, you see these different peaks. So all these different peaks tell us the wavelength of light absorbed by different pigments. Is that clear? You should be able to identify that which peak indicates or which peak is for different which pigment. So let's learn it now. So the first peak that you observe here, this one belongs to chlorophyll A. The first peak you see here is chlorophyll A. Is that clear? Now the second peak, so this is the first peak and this is the second peak and this is of chlorophyll A. Then coming on to the green thing. So the green one, this represents a peak of chlorophyll B and another peak in this region. So on the y-axis, we are taking the absorption of light, while on the x-axis, we are taking the wavelength of light. So first of all, be clear with this also. And then the third one is carotenoids. So we get this kind of for carotenoids. Are these clear? So here we are able to see the absorption spectrum. Now, the point is that the amount of light absorbed by the pigment is directly proportional to the yield or the rate of photosynthesis. Now, the rate of photosynthesis can be studied by action spectrum, and so the second graph shows us the action spectrum for chlorophyll A. Is this clear? So it shows the action spectrum of chlorophyll A. Now, when we overlap the two, action spectrum and absorption spectrum for chlorophyll A, we see this type of graph. We see this type of graph. Now, what is supposed to be understood here is, please see, that action spectrum coincides with the absorption spectrum. Is this clear? And we see the two peaks. One peak is here, another peak is here. One peak is here, another peak is here. Now, this area at which we are able to see these peaks, the two areas, this and wait a second. The two areas, one is this and second is this. These belong to two colors. These belong to two colors, red and blue. Red and blue. Please let me know that these points are clear to you all. Please let me know these points are clear to you all. Yes, King, we will talk about that in a while. So see, this is something we have already discussed. Now coming on to the real thing. See, first of all, you must know that photosynthesis is going to take place in two steps. One is the light reaction and second is the dark reaction, right? Now, why do we call light reaction as light reaction? because here the light is going to play a very important role to cause certain chemical reactions. So we also call it photochemical reaction or light dependent reaction. This is clear. While the dark reaction would be known as light independent reaction because it is not directly dependent. We can't really say it is not dependent because ultimately without light reaction, dark reaction cannot take place. So indirectly, of course, dark reaction is dependent on light, but still because it is not directly dependent, we call it light independent reaction. Also, there are certain new molecules which will be produced. So we also call it biosynthetic reaction. Is that clear? So this is the first followed by dark reaction. Clear? There is no plant on this earth which can perform dark reaction without light reaction, because light reaction is going to produce certain compounds as well, like ATP. It is going to produce NADPH. These are the high energy intermediates. So light reaction is going to produce these things which will be used by the dark reaction. So this produces these high energy intermediates while dark reaction uses them, used in dark reaction. Are these clear? Apart from this ATP and NADPH, light reaction is also going to produce oxygen, and that oxygen will be diffused out of the plants for us. Is this clear? Please tell me that these points are clear to you all. Right? Chief pigment is of course chlorophyll A. Are these points clear to everyone? Now, light reaction will occur in thylakoid because we have just seen that thylakoid is the site, the membrane of thylakoid is the site for these pigments. Yes or no? Also, you can say the stack of thylakoids, which is grana. You will also see that these pigment systems, these photosystems are present. The photosystem is present in stromal lamella also. So that is where light reaction would also take place, while the dark reaction would only occur in stroma. Please tell me that these points are clear. And now I'm going to discuss the light reaction. First, see, it occurs in the thylakoid membrane as well as in the stromal lamella. But the difference is both thylakoid membrane and stromal lamella has different types of photosystems. While in thylakoid membrane, there are two photosystems present, which is photosystem 2 followed and photosystem 1. Both are present. First photosystem 2 will be utilized, followed by photosystem 1. See, the order 1 and 2 is in the order of the discovery. But when it comes to their usage, it is photosystem 2 which is used first, which comes into role first than the photosystem 1. Please tell me that these points are clear. Right? But in case of stromal lamella, we just have photosystem 1. Is that clear? Now let's see how these systems work one by one. Now see, this is the thylakoid membrane. This is the thylakoid membrane. Now on the inner side, see, towards the inner side of thylakoid membrane is present your chlorophyll A molecule or the reaction center. This is the reaction center. And since this is photosystem 2, we are talking about photosystem 2, it is going to be P680. So the chlorophyll A present in photosystem 2 can absorb light up to maximum of 680 nanometer. Are these points clear? Also, see, also the thylakoid membrane has photosystem 1. Now in photosystem 1, the reaction center or the chlorophyll A has a maxima of P700. So it can absorb light maximum up to 700 nanometer. Are these points clear? Now where did I draw all these things? Towards the inner side of thylakoid membrane. Yes? Now, what are the other things present here? Now, towards the outer side of thylakoid membrane is present this PP, pheophytin, right? Also, then within the membrane is present PQ, that is plastoquinone. Then we have cytochrome B6F, and then we have plastocyanin. Is that clear? Now you see the order in which these things are present. So first it is pheophytin, then plastoquinone, then cytochrome B6F, and then plastocyanin, right? And then we have this chlorophyll A again towards the inner side. And then there is another character, another protein, which is ferridoxin, another molecule, FD, also known as ferridoxin. Are you first able to notice where each molecule is present? You should be very, very thorough with the location of these things, first of all. Now, coming to the action, coming to the process. See here. So first of all, we see, just a second. Okay, so we see when the light, see first of all, light is hitting the chlorophyll A. When the light is absorbed by chlorophyll A, being any chemical, so light is energy, chlorophyll is a chemical. This light energy is going to excite the electron of chlorophyll A. Yes or no? So what will happen? The light is falling on chlorophyll A of PS2, as well as light is falling on the chlorophyll of PS1. So when light is absorbed by these chlorophylls and also by the surrounding pigments, ultimately they will also give light to and pass on energy to chlorophyll A. So when chlorophyll A is getting so much energy, the electron of chlorophyll will be excited, and this electron, see, this electron will be taken up by PP, which is pheophytin. Please tell me that this point is clear. So from photosystem 2, the electron will rise up to PP, that is pheophytin, yes, while the electron from photosystem 1 will rise to ferredoxin. So PP, please see here, PP is the primary electron acceptor of PS2. Can I say that? And ferredoxin is the electron acceptor of PS1. Can I say that? Please tell me. Can I say that? Yes or no? Now, let's talk about PS2 first now. So when PS2, the chlorophyll A absorbs light, electron gets excited, it is accepted by PP. So from PP, this electron will be passed on to the PQ. Now from PQ, this will be passed on to cytochrome B6F. Now from B6F, this electron goes to plastocinin. Please tell me that these points are clear. And from plastocinin, this goes directly to the chlorophyll A of PS1. Now, since the chlorophyll of PS1 has already lost its electron, which was accepted by ferredoxin. Now, the electron coming from this electron coming from PS2, this electron coming from PS2 will be able to compensate the electron lost by PS1. Can we say this? Yes or no? And is this much point clear? The electron which was excited from PS1 goes to ferredoxin. Now ferredoxin will pass on this electron to another chemical FNR, which is ferredoxin NADPH reductase. It is an enzyme which is associated with ferredoxin present in the stroma. So see this. Where is FNR? FNR is ferredoxin. What is FNR? FNR is ferredoxin reductase enzyme. Is this clear? It is an enzyme. So FD is going to pass on this electron to FNR, and FNR is going to use this electron as well as H positive ions from the stroma to convert NADP into NADPH2 or NADPH plus H positive. Please tell me that these points are clear. So guys, as I am already going slow, so that in case you have not been able to understand this whole tamasha of electrons, now I think you should be able to understand it. Yes? All clear? Ferredoxin reductase, yes. Points clear to all? Alright. Now see, what happened? So electron excited from PS2, taken up by PP, going through different molecules present in the thylakoid membrane, is given to the PS1. So PS1 lost an electron to FD, but it got compensated from PS2. So it's a very important question. Who compensates the loss of electron from PS1? So it is PS2 which is compensating. Isn't that so? Now see the direction of electrons during the whole process. The direction of, see here, the direction. So the direction is, first it goes upwards, then the electrons flow downwards, and then again the electrons are flowing upwards. So the electron started from here, went upwards, then downwards, and then again upwards. Yes or no? Yes or no? So it is uphill. The flow of electrons is like uphill, downhill, and then uphill again. Please tell me that these points are clear to all of you. These points are clear to all of you, right? Now, because, so again, I'm telling you what is the direction? Uphill, downhill, uphill. Udu. Can you remember Udu? So Udu is flying. So electrons are flying upwards, downhill, and uphill. Is that clear? All right. So now this can be regarded as N also. So somebody can see N being formed here, but the scientists saw Z here. So they said it is a Z scheme of electron flow. So scientists called it Z scheme of electron flow. Are these points clear? So Z scheme is because of the shape in which the electrons are moving uphill, downhill, and uphill. Clear to everyone? All right. Now see here. The electron which was lost from PS1, as I said, is coming from the PS2, right? The electron from PS2 is going to compensate that electron. But what about the electron lost here? Electron which has been lost from PS2, what about that? So if there is nothing to compensate this loss of electron, ultimately the chlorophyll, the chemical, will cease to exist, right? So there is something happening on the inner side of the membrane. So what is that something? So on the inner side of the membrane, guys, there is this complex present. This is known as water splitting complex. What is this known as? Water splitting complex. Where is this water splitting complex present? This water splitting complex is present on the inner side of the membrane. Are these points clear to all? Please tell me. Right? Now this water splitting complex, as the name says, is going to split water. So when light falls on chlorophyll A and the electron is lost, simultaneously water is also going to split here to form H positive ions and oxygen molecules, and electrons are lost. Right? Now the electrons formed here, these are going to compensate. These electrons are going to compensate for the loss of electron from chlorophyll A of PS2. Please tell me these points are super clear. They should be super clear. I can't accept anything less than that. Yes, guys, give me a thumbs up that these points are super clear to you. Right? Are these points clear? Perfect. Moving ahead then. So, PS2 is getting compensated by the splitting of water, and PS1 is getting compensated from PS2. These are the two very important lines. Clear? Perfect. Now also, see, this is the lumen. This is the thylakoid lumen. Everything that we have studied so far is happening in the membrane of thylakoid. Yes or no? Right? Now what is happening in the thylakoid? So when water is getting split, these H positive ions are getting collected in the thylakoid membrane. Yes or no? They are getting collected. So this leads to high concentration of H positive ions. Is this point clear? Also guys, please note. So PP, pheophytin, was an electron acceptor. But when it was going to pass on this electron, when PP had to pass on these electrons, TC, when PP had to pass on these electrons to PQ, plastoquinone was not a very kind guy. Why? Because plastoquinone said, see, it's not my work. I'm not an electron carrier. Remember, plastoquinone is not an electron carrier. Plastoquinone is a hydrogen carrier. So you are asking a hydrogen carrier to pass on the electrons. So what happens? Plastoquinone is not going to do this for free. What's plastoquinone going to say? Okay, if you want me to do a work for you to pass on your electron to cytochrome C, I am going to do my work at the cost of that. So you'll have to let me pass these electrons. Let me pass these H positive ions from the stroma into the lumen at the cost of transferring your electron to cytochrome C. Are you getting that? There are people who don't do anything for free. So if you ask for a favor, they'll say, this is not my work. You let me do this, then I'll let you do your work. right? Then I'll do your work. So PQ is one such guy. PQ is one such guy. So in order to pass on the electron from PP to cytochrome C, B6F, cytochrome B6F, PQ, plastoquinone, is going to transfer, H positive ions from stroma into the thylakoid membrane. So this also contributes to high H positive ions in the thylakoid membrane. Are you getting that? So there are two reasons because of which H positive ions are increasing in the thylakoid lumen. One is due to splitting of water, and second, it is because of plastoquinone, which is a hydrogen carrier, and it transfers these H positive ions actively. So using ATP, they are going to transfer these H positive ions into the lumen. So what happens? So there is a gradient set. Please see, there is a gradient. Gradient means difference being set here. Low H positive concentration on the stromal side and high H positive concentration in the lumen. Are these points clear? Please let me know. So guys, this gradient is of huge importance. Now see, there is an ATP synthase. There is this molecule ATP synthase present in the thylakoid membrane. Now, ATP synthase is an enzyme which, as the name says, is going to synthesize ATP. Now, ATP synthase has two components. This component is known as CF0. C stands for chloroplast. Chloroplast CF0, yes. And there is second component which is CF1. CF1. So, this is CF1. Right? CF1 is towards the stromal side. So see the location also. Where are these things present? So CF1 is towards the stromal. Not is buried in the, it is embedded in the, where? It is embedded in the, CF0 is embedded in the thylakoid membrane. Embedded, and this one is towards the stroma. Please tell me that these points are clear to you all. Yes or no? Is it lumen of membrane that's higher, or is there no difference? Grumpy, see, told you there is high concentration of H positive ions in the lumen. Okay, and lower on the stromal side. And you know the difference now. Why are there high concentration of H positive ions in the lumen? One, because of splitting of water. And second, of plastoquinone, which is pumping in. We say pump because it is at the expense of ATP, energy. And why is H positive ion concentration low on the stromal side? One, because plastoquinone is pumping it in. And second, for low concentration, it is FNR which is responsible. Do you see this FNR is using the electron and H positive ion? So the remaining H positive ions which were left over in the stroma are getting used by FNR to produce NADP, H positive and H positive. Please tell me that these things are clear to you all. Are these all points clear? Yes or no? Loud and clear? Perfect. If it is loud and clear, then that's exactly what I'm trying to do. All right. Then see, guys. So CF0, which is embedded here, and CF1, which is on that stromal side, this is a complete ATP synthase. This is a complete enzyme, right? Now see what's happening here. Now, clearly, okay, yes. Now you will see. So H positive ions being more in the lumen and less towards the stroma. So they will easily move from high concentration to low concentration, yes or no? Please tell me whether this will happen or not. So H positive ions are more in the lumen and less towards the stroma. So from high to low concentration, the H positive ions will start moving. Please tell me whether this will happen or not. But obvious? Yes, H positive ion concentration, H positive ions move from lumen to stroma. Yes or no? From high concentration to low concentration. If there is a gradient set, how will things move? They will automatically move along the gradient. So when they are going to move along the gradient, we don't need any energy. What type of transport is this? This is a passive transport. This is a passive transport. Yes or no? And such passive transport is known as diffusion. Yes or no? But this diffusion is slightly different because this diffusion is occurring through a particular protein. So CF naught is CF naught present in the membrane is acting as a transmembrane protein. It is acting as a channel. So there is a channel for the diffusion. So whenever diffusion occurs through a channel, we call it facilitated. With the facility of a protein, we call it a facilitated diffusion. It is a passive transport, but of which type? Facilitated diffusion. Yes or no? Right? So guys, the H positive ions move from lumen to stroma through passive transport, through facilitated diffusion, and during this transport, now this is very, very important. So during this transport, during the transport, what happens? For passage of every 4 H positive ions, 1 ATP is produced. For every transport of for the transport of every 4 H positive ions, 1 ATP molecule is produced. So the formation of ATP is known as phosphorylation. ATP formation is known as phosphorylation. Yes or no? And since this phosphorylation is occurring during the light reaction, it is known as photophosphorylation. Can we say that? Yes or no? Also, note down the electron which was once lost from chlorophyll A, PS2, never came back. It moved further to PP, to PQ, to cytochrome B6, to PC, from PC to PS1, and then FT, and went into the stroma and became NADPH. Yes, it formed NADPH. So the electron once lost never came back. They were compensated from one or the other way, in one or the other way, but they never came back. So can we say that this Z scheme is or represents non-cyclic electron flow? So can we call such photophosphorylation as non-cyclic photophosphorylation? Phosphorylation means ATP production. We call it photo because it occurs during the light reaction. We call it non-cyclic photophosphorylation because it occurs during the non-cyclic electron flow. Can we say all these things? Please tell me. Have you understood the whole mechanism? So see, so we have talked about the non-cyclic photophosphorylation. But remember, there is one more way. So these are the full forms. These are the different natures of these compounds present in the thylakoid membrane. Already told you PP towards the outer side, PQ within the thylakoid membrane, FT and FNR towards the outer side, NADP in the stroma. And these are the full forms. And what are their natures? Pheophytin is the primary electron acceptor of PO2, PS2. This is electron acceptor of PS1. Plastoquinone is the H positive carrier, and that is why it was not transporting electrons all for free, right? While cytochrome B6F and plastocyanin are electron carriers. Please tell me all these points are super clear. See, what all happened during light reaction? What all happened during light reaction? Number one, absorption of light by chlorophyll A, that is the reaction center, followed by photoexcitation of electrons from the reaction center. That means loss of electron took place from chlorophyll A, both from PS2 and PS1. Yes. So we see that from P680 accepted by PP, PQ moving to PQ, then cytochrome B6F, then PC, right? And from P700 it went to FD, then electron from FD went to FNR. FNR converts NADP to NADPH positive plus H positive by taking electron which was coming from behind, and H positive were taken from stroma. Please tell me all these points are clear to you. And also, water splitting took place to compensate for the loss of electron for PS2. Yes. So water splitting takes place. So we know that H positive remain in lumen, oxygen is released in the atmosphere, and this electron will compensate the PS2. Please tell me these points are clear. So another thing that happened during this light reaction is ATP production. We have seen that non-cyclic photophosphorylation has already been seen. So when the non-cyclic photo and this is known as chemiosmosis hypothesis actually. So see, let's see this. So when electrons are flowing from through CF1 and flowing from lumen to stroma, This is known as chemiosmosis. See, what is osmosis? Osmosis is the flowing of water, right, to from high concentration to low concentration. That is osmosis across a membrane, right? So here, instead of water, it is the H positive ions which are flowing. So we called it chemiosmosis. We called it chemiosmosis. So chemiosmosis hypothesis took place for the ATP production. Are these points clear? So there was a proton gradient created, and this proton gradient ultimately helped in ATP production. Now, very, very important question here. Chemiosmosis hypothesis. It is involved with movement of H positive ion from high concentration to the low concentration. Where is high concentration? That is in lumen. Where is low concentration? That is stroma. So, since it is moving from high to low, it is a passive transport. Which method of passive transport? Facilitated diffusion, because there is requirement of a carrier protein, which happens to be CF0. So what are the requirements? Very, very important thing. We need a proton gradient for the protons to flow. Gradient means difference. If there will be no difference, protons won't flow. So first of all, we need proton gradient. Then we need a membrane across which they can flow. Third, we need a pump, right? And fourth, we need this ATP synthase. This enzyme. So four things, four requirements for chemiosmosis hypothesis are these, and this is a very, very important aspect. We have already seen a PYQ on this topic. This is a PYQ. Is this clear? Are these points clear? All right. So ATP synthesis, we have already understood that this ATP synthesis has two components, CF0, CF1, and how does it help? For every 4 H positive ions passing through, 81 ATP molecule will be produced. Now see, the second part of light reaction. So while we have seen things happening in the thylakoid membrane already, which has PS2 as well as PS1, now there is a photosystem present in the stromal lamella also. So there is a photosystem present in the stromal lamella also. So this is a stromal lamella. This is a stromal lamella. Stromal lamella is the membrane which connects the two thylakoids or the two grana. Yes or no? Now, please see that unlike the thylakoid membranes which had both photosystems, photosystem 1 and photosystem 2, this one has only PS1. This only has photosystem 1. There is no photosystem 2. So remember, in stromal lamella, photosystem 2 is absent. Is this clear? See the difference also. So what will happen? So the electron from the photosystem 2 will jump to ferredoxin. That is what was happening previously also. So electron acceptor of PS1 is always ferredoxin. It's the same story here. So ferredoxin is the electron acceptor of PS1 here. But unlike in in thylakoid membrane, where ferredoxin was passing on that electron to FNR, here that will not happen. Why? The FNR is also absent. If there is no FNR, NADP cannot be converted into NADP, H positive and H positive, right? So what happens? The electrons, see here, the electrons which jump to FD, they will come back to the molecules present in the membrane PQ, and from PQ again cytochrome B6F. So the same electron will be coming back indirectly to the chlorophyll, which once jumped out or got excited and jumped out of it. The same electron will jump back. It will again come back. So that is why we call this as cyclic electron flow. So the difference being, there is no PS2, there is no FNR. So we don't need an outsider to compensate for the electrons lost, because the electron once lost is ultimately going to come back. Yes or no? So that is what makes a cyclic electron flow. Are these points clear? Yes or no? So I've already written, see, FNR is absent. So because there is no FNR, there will be no NADPH. No NADPH. Clear to all? Now since there is no PS2, there will be no water splitting. And moreover, it is not even needed. Since there is no water splitting, there will be no oxygen. During the cyclic electron flow, guys, there is no oxygen produced, there is no NADPH produced. But again, there will be transport of H positive ions. So ATP production will take place. So ATP production will take place. Is this point clear? ATP production will definitely take place. So these are some points of differences between non-cyclic and cyclic. So the ATP production which occurs during this phase, see here, the ATP production which occurs during this phase will be known as, see here, told you. All right, let's move again. So during light reaction, ATP production will take place, right? That is why we call it photophosphorylation. Yes or no? Photophosphorylation, right? Now the photophosphorylation, if it occurs during Z scheme, which is a non-cyclic one, will be known as non-cyclic photophosphorylation. And the one which occurs during cyclic flow will be known as cyclic photophosphorylation. So there are two methods of ATP production during the light reaction. Please tell me if these points are clear. So in the light reaction, in the Z scheme, along with ATP production, we know oxygen is also produced and NADPH is also produced along with ATP. While in cyclic, it is only ATP that is produced. Rest of the two things will not be produced. So these are the things that is happening here. Read these lines, and then now coming to dark reaction. So the products of light reaction will be utilized here. Which all products? One is ATP and second is NADPH. They will be utilized here, right? They are used here, used in dark reaction. That is why without light reaction, dark reaction can never take place. Okay, I told you this one is known as biosynthetic pathway or phase, because some biosynthetic molecules, organic molecules will be synthesized. Also known as light independent reaction because it is not directly dependent, although light independent reaction is not a very good name for it. It occurs in the stroma, and pathway was studied by Melvin Kelvin, an American biochemist. And we also call this pathway as C3 cycle. Why? Because the first molecule formed during the cycle would be a 3-carbon compound. Is that clear? Okay, coming on to the C3 cycle. See here, C3 cycle. So now this cycle is going to occur in the stroma. See here. So ATP from thylakoid has reached the stroma. It has reached the stroma. Now in stroma is present, a molecule called RUBP. See, it is very important that you must know that which compound has how many carbon atoms. Is that clear? So RUBP is ribulose bisphosphate and it is a 5-carbon compound. Now 5-carbon compound, it accepts 1 carbon dioxide. It accepts 1 carbon dioxide molecule. So 5 plus 1 becomes 6. 5 plus 1 becomes 6. Yes or no? Now RUBP is going to accept CO2 in the presence of enzyme Rubisco. Now since there is addition of CO2 in RUBP, the process is known as carboxylation. Yes or no? Now the molecule formed here should be a 6-carbon compound, right? There is 5 and this is 1, total 6. But instead of forming a 6-carbon compound, the reaction is going to produce 2 molecules which are 3-carbon compound each. So the molecule will be PGA, phosphoglyceraldehyde or glyceric acid. This PGA, the 2 molecules will be formed. So 2 molecules of 3-carbon compounds is equal to 6. So that's how it gets compensated. So 1 molecule of RUBP joins 1 molecule of CO2 and results in the formation of 2 molecules of PGA. Is that clear? Now each molecule of PGA will undergo reduction. Is that clear? 2 molecules will undergo reduction. Now each molecule will require 1 ATP and 1 NADPH. And now since we have 2, so we'll need 2 ATP and 2 NADPH. So this step of reduction, this is A, carboxylation. Second is reduction. So during reduction, 2 ATP and 2 NADPH are used for the reduction of 2 molecules of PGA. Please understand these points are clear. And it results in the formation of triose phosphate. Again, 2 molecules. Is it clear? Again, 2 molecules. Again, 3C. And from this 3, the 2 molecules of 3 carbon compound, triose phosphate, the 2 into 3 is 6. Now, what will happen? One carbon will move out, and rest of the 5 carbons will result in the regeneration of RUPP. Are these points clear? Now this step is known as regeneration and it utilizes one molecule of ATP. Is that clear? So first of all, in C3 cycle, it occurs in 3 steps. One is carboxylation, followed by reduction, followed by regeneration. First question. These are all PYQs. This is one of the most important topics. And followed by regeneration, right? Now, during this whole process, how many ATPs are being utilized? 2 here, 2 during the reduction and 1 during the regeneration. So in total, 3 molecules. And how many NADPH are being used? 2 NADPH molecules, yes or no? Clear? So one cycle of C3 cycle utilizes how much? One cycle of C3 utilizes 2 molecules of NADPH and 3 molecules of ATP. Are these points clear? And after every C3 cycle, we have one carbon outside. Now, if we have to produce C6H12O6, that is one molecule of glucose, tell me, if we have to produce one molecule of glucose, how many such carbons are needed? We need 6 such carbons. How many cycles will take place? 6 C3 cycles will take place. 6 C3 cycles will take place to form one molecule of glucose. Yes or no? Because each cycle is releasing one carbon. We need 6 carbons. So you need to repeat the C3 cycle 6 times to form one molecule of glucose. So it means during 6 cycles, that is for the formation of one glucose molecule, how many ATP and NADPH will be used. So how many ATP and NADPH will be used? It will use. So 2 into, just multiply it with 6. 6 to the 12 NADPH and 18 ATP molecules will be used for the formation of one glucose. So similarly, if the question is how many ATP and NADPH are used for 3 CO2 molecules for fixing, we call this fixation, right? For fixing 3 CO2 molecules, how much ATP and NADPH will be used? Can you answer this question? If you can answer this question, your NEET will be clear. Those of you who will watch this session later on, please don't forget to leave the answer in the comment box, and rest of you answer in the chat. answer this in the chat. So guys, this is the full form RUBP. It is a primary CO2 acceptor for C3 cycle. For C3 cycle, primary CO2 acceptor is what? Wait. The primary CO2 acceptor is RUBP. And then the enzyme used is Rubisco. We know the full form. Carboxylase and oxygenase, the most abundant enzyme in the biosphere. We have just learned it yesterday. Right? Good, guys. Very good. Very good. And what is PGA? Phosphoglyceraldehyde or glyceric acid. And this is a 3-carbon compound. You should also know how many carbon atoms are present in each molecule. Is this clear? All right. So, light reaction produced ATP and NADPH, and CO2 was not even used during the light reaction. And these were used. And during the dark reaction, this ATP was used, and as a result, one glucose molecule was produced. Please let me know that these points are clear. So when ATP was used, it resulted in the formation of ADP. NADPH positive was used to form NADP. Now these will be recycled to the light reaction. Remember, the PS1 FNR is going to convert NADP back into NADPH. Remember that? Yes or no? It will be formed. This will be reformed into NADPH. Clear to everyone? Please let me know these points are clear. So we already know about Rubisco. It has a dual nature. Okay. So it has carboxylase activity when CO2 is more. If oxygen concentration is more, then instead of carboxylation, it will start performing oxygenation. And that also happens. Now see, what will happen? See, we have already seen the C3 cycle. But in case, see, let's talk about this situation when oxygen is in higher concentration. Now Rubisco is going to change its favorism. So, okay, so instead of Rubisco adding carbon dioxide, instead of RUBP getting added with CO2, RUBP will now add oxygen in the presence of Rubisco. So oxygenation would take place and it will result in the formation of one 2 carbon compound and one 3 carbon compound. Is that clear? Now this 3 carbon compound is again PGA. It is the same PGA which was produced in C3 cycle. But this 2 carbon compound is phosphoglycolate. Phosphoglycolate. Now this phosphoglycolate will further get into a different metabolic pathway. But as a result, just know, see, as a result of C3 cycle, we found glucose. We produced glucose. So this is the benefit of C3 cycle. C3 cycle was using ATP, it was using NADPH, but what did we get out of C3 cycle? We got food. We got glucose. But as a result of C2 cycle, we get zero, we get anda, we get nothing. So C2 cycle is called a wasteful cycle. Neither does it produce ATP, nor does it produce NADPH, nor does it produce glucose. So what is the fun of a process going on and utilizing ATP and everything and not giving us anything in return? Is that clear? So see, that is why we call it a C2 cycle, because one of the 2 carbon compounds is being produced here, which is phosphoglycolate. And it is a wasteful process because there is no useful product being produced here. No glucose, no oxygen, no ATP, no NADPH. So it is a wasteful process. Instead, it wastes our resource. The same RUBP which could have added CO2 is now adding oxygen and giving us nothing. Are these points clear to everyone? Please let me know. So now gradually there was an evolution of another category of plants known as C4 plants. They had a special anatomy which will enhance C3 cycle and it will decrease C2 cycle. Okay, so from C3 plants got evolved, gradually it evolved into C4 plants. So there are few plants like C4 plants which are mostly monocots. So C4 plants are mostly monocots, but then there are few dicots also which are C4 in nature. For example, maize, barley, sorghum, sugarcane, these are all monocots. But remember, rice is also a monocot, but it is not a C4 plant. So it's not mandatory that all monocots will be C4 plants. Okay. Now, C4 plants being C4, it doesn't mean that C3 cycle will not take place. See, let me tell you two things. There is not even a single plant performing photosynthesis in which light reaction is not occurring. Light reaction is mandatory, without which dark reaction cannot occur. Secondly, there is no plant which is photosynthesizing, which is making food, but it is not doing it through C3. Remember, glucose will ultimately be produced by C3 cycle only. Are you getting that? Are you getting that? But then what is the significance of C4? Let's see this. So in both C3 and C4 plants, C3 cycle will take place because it is during C3 only that glucose is getting produced. Now see here, what is this doing? In C4 plants, there is a special Krebs anatomy. Okay, there is a special Krebs anatomy. All right. Okay. Now see the Krebs anatomy. What is Krebs anatomy? So around the vascular bundle, there are special bundle sheath cells present. Now these bundle sheath cells, you must understand the nature. They are thick-walled cells. So once the gas is produced within bundle sheath cell, it will not be able to escape because the wall is thick. Wall is very thick. Is that clear? And So see, these are the bundle sheet cells, right? Now in C4 plants, what happens in C4 plants? There are two cells which are going to participate. One is the mesophyll cell and second is the bundle sheet cell. While in C3 plants, it is only the mesophyll cells that will participate. Is that clear? Now, in C4 plant C, the mesophyll cell, instead of having RUPP, has PEP. It has PEP. Now, PEP is going to accept CO2. Instead of RUPP, who is the primary CO2 acceptor in C4 plants? It is PEP, phosphoenol pyruvate. Clear? In the presence of PEPcase, now it is not Rubisco, it is PEPcase which is getting used for carboxylation. So the CO2 molecule enters, one CO2 molecule enters in the mesophyll cell. Now these mesophyll cells will not have Rubisco, while in C3 plants, mesophyll cell has Rubisco. But in C4 plants, mesophyll cell does not have Rubisco, instead it has PEP. Now CO2, PEP is a 3-carbon compound. It is a 3-carbon compound. 3 carbon plus 1 CO2 will add up to form 4-carbon compound, which is oxaloacetic acid. And because the first stable compound is a 4-carbon compound, that is why we call it a C4 cycle or a C4 plant. Right? Now this oxaloacetic acid will change into aspartic acid or malic acid, and both are 4-carbon compound. In some plants, it will form aspartic. In others, it will form malic acid. Either way, both are 4-carbon compound. Now, this malic acid will enter into the bundle sheet cell. So, do you see the connection here? So, here there is no Rubisco. Instead, it has PEP. And from here, the malic acid produced is transported into the bundle sheet cells. Are you getting that? Through plasmodesmata. Right? Now, malic acid or aspartic acid, when it reaches the bundle sheet cell, so this malic acid will lose one carbon dioxide. So that means decarboxylation will take place. Is that clear? So decarboxylation will take place. So from 4C, it will become a 3-carbon compound, which is a pyruvate. So from malic acid, it will form pyruvate. Is that clear? So pyruvate is a 3-carbon compound. This pyruvate goes back into the mesophyll and will help in regeneration of PEP. Is that clear? And during regeneration, one ATP molecule will be converted to one AMP, so which is equivalent to 2 ATP molecules. Just understand. This is for one carbon dioxide getting fixed. Are these points clear? Now, talking about the CO2 which was released from malic acid. So, malic acid underwent decarboxylation. So, one CO2 was lost here. Now, the CO2 lost or produced here in the bundle sheet cells, see, I told you, they have very thick wall. Now, this CO2 will not be diffused out of the wall of bundle sheet. It will stay within the bundle sheet cell. Now, when the CO2 is being produced regularly as a result of this cycle, C4 cycle, this is a C4 cycle, right? So, and this CO2 is filling the bundle sheet cell. So bundle sheet cell becomes a carbon dioxide chamber. Can we say that? So bundle sheet cell becomes a carbon dioxide chamber. So the CO2 concentration is much higher than oxygen. So what will happen? Rubisco will now favor carboxylation instead of oxygenation. So now here the Rubisco present, here is Rubisco. The Rubisco present here will fix carbon dioxide and not oxygen. And as a result, we know glucose will be produced. Are these points clear to everyone? Please let me know that these points are clear to all. Please let me know these points are clear to all. Right? Now let's see the expense of energy here. So see, during one C3 cycle, let's talk about it here. During one C3 cycle, we know there are 3 ATP molecules and 2 NADPH molecules It uses one C3 cycle uses 3 ATP and 2 NADPH, and one C4 cycle was using how many? 2 ATP molecules during regeneration of PEP. Yes. So how many will 6 cycles of C3 and C4 use? So in all, so 2 and 3, 5 ATP. 5 ATP and 2 NADPH were used. So 6 cycles will utilize how many? 30 ATP molecules and 12 NADPH. So C4 cycle, overall C4 plants, see, C4 plants will have C3 cycle as well as C4 cycle. But first C4 cycle followed by C3 cycle. C4 cycle in the mesophyll cells and C3 cycle in the bundle sheath cells. But overall they are using more ATP. But because C2 cycle will not take place, take place, even though ATP is used more, still the yield will be higher in C4 plants. Please tell me that these points are clear. So guys, now I am leaving you with these tables. These are very, very important and we have already discussed that. So don't forget to download the notes and read this. Then factors affecting the rate of photosynthesis. One is light, very abundant. Sun is a source. So light is rarely a limiting factor, but in case of shade loving plants, it can become a limiting factor. And dense forest, because shade loving plants require less light, and in dense forest less amount of light will reach the surface at the bottom. Is that clear? So that is when light can become, otherwise normally light is not a limiting factor. Then water indirectly affects the rate of photosynthesis. How? Because water stress can close the stomata. If the stomata will be closed, then CO2 will also not be available. CO2 enters through stomata. Leaves will wilt and surface area will also reduce. Again, less light will be trapped by the leaves. So not directly, but water affects indirectly. Then we'll talk about temperature. So temperature has to be optimum. Okay. The best temperature for C3 plants is between 20 to 25 degrees Celsius, while C4 plants can work at a higher temperature, that is around 40 degrees Celsius or little above. Is that clear? Now, how does temperature really affect? So temperature doesn't affect the light reaction. Temperature is going to affect the dark reaction, because dark reaction is enzyme based. And we know that enzymes are temperature specific. Okay. So dark reaction is affected. Then coming on to the CO2 concentration. So of course, CO2 concentration will affect rate of photosynthesis. Now, usually we might think the more the CO2, more will be the glucose form, but that's not the case. So the atmospheric concentration of CO2 is between 0.03 to 0.04. But maximum percentage of CO2 which the plants can tolerate is only up to 0.05%. If you go beyond 0.5% CO2, then CO2 becomes toxic. So do you understand? It's not the case that, okay, let the pollution increase, at least plants are happy. No, plants are not happy by getting so much of CO2. Even CO2 becomes toxic for the plant. They cannot tolerate more than 0.5% of CO2 in atmosphere. Is this point clear? Is this point clear to everyone? So see here, see these lines are very, very important, very, very important here. So the C3 plants, okay, wait. C3 plants respond differently to CO2 concentration. What you must note here, the C3 plants, yes, C3 plants show saturation at C4 plants will show saturation at 360. C4 will show saturation at 360, while C3 will show saturation at 450. Please tell me that these points are clear to you all. Please tell me these points are clear to you all. And then we have this very important paragraph that has already been discussed. So light shows a linear reaction at low intensity. Light shows a linear reaction. So till there is low intensity, we get a linear curve here. So this is happening because light intensity is low. So we get a linear relationship, right? At point B, the dependence on light goes on decreasing. See, at higher light intensity, the rate does not show further increase. Because why it is slow here? Because the other factors become limiting. And then beyond a certain point, it becomes straight to the x-axis. Why? Because here light saturation plateau phase is because light saturation has occurred. So light saturation occurs at 10% of the full sunlight. Now at this phase, light is not the limiting phase, anything else can be. Is this point clear to all? Please let me know that these points are clear. So these are the experiments that we have already studied. See, how do we understand that chlorophyll is important for photosynthesis? By studying it through the variegated leaf experiment. So you take a multicolored leaf, but we'll see that photosynthesis happens only in the green parts, and because photosynthesis is occurring in the green parts, that is why only the green part will give a positive iodine test. So in the presence of starch, iodine changes to from yellowish brown to blue black. So that blue black color will only be noticed in the green parts. Is that clear? Then KOH. This experiment will prove that carbon dioxide is important. Carbon dioxide is important for photosynthesis. See, you put one leaf in a beaker which contains KOH. KOH will absorb CO2, and after a few hours we will see that this plant has not photosynthesized, so it will not give an iodine test. Is that clear? Because no CO2, no KOH, no photosynthesis, no starch, and iodine doesn't change its color. Is that clear? Then coming on to next is the Joseph Priestley's test in the bell jar experiment, where he explained that it changes the what was the conclusion. So burning candle and animal, they damage the air. So plants, so this plant, it improves the quality of air which was changed by the burning candle and the animal, right? See this. Now engine house proved that light is essential for photosynthesis. He also experimented with this aquatic plant. So when the aquatic plant was placed in sunlight, it was producing these bubbles, which were found out to be oxygen. But when the same setup was kept in dark, these oxygen bubbles were not produced. Then we have Julian von Sachs. He proved three things. Number one, that green parts produce glucose. Glucose is stored as starch, and then green substance is located in chlorophyll. Are these clear? And Engelmann, he did this prism experiment. With the help of prism, he splitted the white light into Vibgyor, and then exposed the green algae to these different lights. And it was observed that Cladophora, the green algae, it showed maximum growth or multiplication in the red and blue regions. Are these points clear? While in green, it was a least. So least in green. So it means maximum photosynthesis will occur in blue and red, and the least would occur in green light. Is this clear? Then Cornelius Van Niel showed that the source, if the source of hydrogen donor, if the hydrogen donor is water, it will lead to oxygen production. But if hydrogen donor happens to be H2S, it will lead to this production of sulfur. Is that clear? So hydrogen comes from a suitable oxidizable compound and reduces CO2 to form carbohydrates. But during that process, this was also inferred. Clear? He only inferred that source of oxygen is water and not CO2, and to prove this, he did a radioisotopic experiment. Are these points clear to everyone? Please let me know, guys. Please let me know. All right, and then moving on to the last chapter of plant physiology, which is respiration in plants. What is respiration? Breakdown of food to release energy. It is a catabolic process, but we see that during this whole entire process, there are some steps which involve anabolism also. So overall, it is an amphibolic process. Overall, it is an amphibolic process. It occurs in each and every cell because ATP has to be produced by each and every cell on their own. Now, the fuel used, the compounds which we use, the substances that we use to produce energy, they are known as respiratory substances, which happens to be carbohydrates, fats, proteins, and organic acids. But the most used or the usually used substrate is, of course, carbohydrate. This one is the usually used one. Clear? All these points, and these are PYQs. Now let's see the basic process. So what happens? In the cell will enter oxygen and glucose. Okay. Now one glucose molecule, which is a 6-carbon compound, it will break down by the process called glycolysis. Now this glycolysis is itself a 10-step process. We'll discuss these 10 steps. But let's see the basic. Glucose undergoes glycolysis in 10 steps to form 2 molecules of pyruvate. Why? Because pyruvate is a 3-carbon compound, so according to the law of conservation of mass. Now during the glycolysis, oxygen is not used. So this is an anaerobic part of the process and occurs in the cytoplasm. It occurs in the cytoplasm. Now pyruvate can show 3 different fates. Okay. Now see, if there is lack of oxygen, that means anaerobic, then this pyruvate will remain in the cytoplasm. This is cytoplasm. This is all cytoplasm. If there is lack of oxygen, then pyruvate remains in the cytoplasm. Now, depending upon the cell, it will either form ethanol plus two molecules of carbon dioxide, two molecules of ethanol, and two molecules of ATP. Clear? And if it happens to be in our muscle cell, when we are exercising or there is too much of physical activity and lack of oxygen occurs in the muscle, it will lead to the formation of lactic acid. But see, during lactic acid formation, CO2 is not produced. Here, CO2 is not produced because lactic acid itself is a 3-carbon compound. Are these points clear? So energy produced in both the cases is just 2 ATP. Very less amount of energy is produced here. But now, if oxygen is available, and then in that case, pyruvate will enter the mitochondria. The first of all, pyruvate on entering the mitochondria will change into 2-acetyl coenzyme A. Now see, this pyruvate is a 3-carbon compound. Acetyl coenzyme is a 2-carbon compound. So it means there is one less carbon dioxide. Now that one less one less C is lost in the form of CO2. So this reaction is known as link reaction. This reaction is known as link reaction, which converts 3-carbon compound pyruvate into 2-carbon compound acetyl coenzyme A, and one CO2 is released. Are these points clear? Now, where is it occurring? It is occurring in the mitochondrial matrix. This is all mitochondrial matrix. Please tell me that these points are clear. Now, this 2-acetyl coenzyme, so 2 molecules of acetyl coenzyme A is produced, right? Now, each acetyl coenzyme A will enter the Krebs cycle, right? And as a result, and after Krebs cycle, there will be electron transport system also. So as a result, we will have carbon dioxide plus water, and lot of energy will be produced. Now, how this happens, let's see it one by one. Now see, during the glycolysis reaction, it is also known as Embden-Meyerhof and Paranas pathway, EMP, breakdown of glucose. This breakdown is partial breakdown. It is a partial breakdown. Complete breakdown means it is forming oxygen and water. Now that only happens during the aerobic respiration. Otherwise, during lactic acid formation or alcohol formation or even this glycolysis, it is complete. It is just partial breakdown of glucose. Occurs in cytoplasm, 10-step process, and studied by these 3 scientists. Now see, we all have heard money makes money. Similarly, in order to produce energy, you need to put in some energy. So in glycolysis also, in the initial phase, some ATP will be used. How much ATP? 2 molecules of ATP will be used. But in the later part of the glycolysis, we'll see the 4 molecules of ATP will be produced. Are you getting that? So first you put in ATP and then you produce more ATP. Also, during the glycolysis, NADH will also be produced. How many molecules? 2 molecules. And remember, you'll see it in this session that NADH is equivalent to 1 NADH is equivalent to 3 ATP molecules. So 2 will be equivalent to 6 ATP molecules. Are these points clear? So ultimately, during one cycle of glycolysis, what happens? So how much ATP is produced? 10. So 4 ATP here and 6 ATP indirectly. So overall, 10 molecules of ATP are produced. And if we subtract the 2 ATP which were used up, so the net gain of ATP during glycolysis is 8 ATP. But if it asks you how many ATP were produced directly, how many ATP are produced directly, then in that case, it has to be 4 minus 2, that is 2 ATP only. Okay? Because this conversion of NADH into this ATP, it occurs through a different pathway, which only occurs in the aerobic process. Is that clear? So the ATP produced directly are 2 ATP, but net gain should be 8 ATP. Are these points clear? Please let me know. Now we already know from the previous chapter that ATP formation is known as phosphorylation. Now this is not for photophosphorylation because it doesn't utilize light energy, or it doesn't occur in the presence of light. So this is simply for phosphorylation. Now phosphorylation can be of two types. One will be substrate level, second will be oxidative phosphorylation. Now what do we understand by substrate level? See, there is one molecule which has phosphate, right? And if this substance becomes this AP, there is this phosphate molecule attached to A. Now, if I subtract phosphate from here and add it to ADP, it will form ATP. Is that clear? Just understand the basic. So when we are taking phosphate from a substrate, this is a substrate, right? And this is a product. So when we are taking out phosphate from a substrate and adding it to ADP to form ATP, such ATP formation is known as substrate level phosphorylation. Are these points clear to all? Right? Okay. So for example, when phosphoenol pyruvate changes into pyruvate, so phosphate from, this is PEP, right? So the phosphate from PEP is taken out, added to ADP to form ATP. This type of phosphorylation is substrate level phosphorylation. While in oxidative phosphorylation, there will be oxidation reaction during which ATP will be produced. Now that happens during ETS. So that will be known as oxidative reaction. So now see, you have to learn the process of glycolysis very thoroughly, because you will directly get questions that during which reaction ATP is being used or ATP is being produced and so on. So which of the following steps shows substrate level phosphorylation? These are the type of questions you get. So here we see there is 1,3-BPG. There are two phosphate molecules. Bis phosphoglycerate means two molecules of phosphate, and it becomes 3-PG phosphoglycerate. It means one phosphate has been taken out and given to ADP to form ATP. So this is known as the substrate level phosphorylation. Are these points clear? Please let me know. So make sure that you learn that 10-step process. See, how do we learn that? Also see this. So we have a mnemonic. Good girlfriend fills glass by 3 pg and 2 peg and plays ping pong. So this is the not my mnemonic, but you can easily find such mnemonics. But it helps to remember, right? So first thing is glucose. Then glucose will use ATP. Now see here. What are the steps which use ATP? So we give ATP here. Glucose uses ATP, takes out one phosphate and becomes glucose 6-phosphate. Is that clear? Now glucose 6-phosphate will undergo isomerism and form fructose 6-phosphate. Is this clear? Then fructose 6-phosphate will again take one phosphate, from where again ATP will come in. So these two steps, glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6-bisphosphate, are using ATP. Are you getting it? So these are energy utilizing steps. Right? So how many molecules of ATP have been used? 2 ATP have been used. Are these points clear to all? Please let me know. Now, this phosphate 1,6-bisphosphate will undergo splitting. So see, this is also, let me mark it here. This is also 6-carbon compound. This is also 6-carbon compound. Fructose is also 6-carbon compound. This is also a 6-carbon compound. But then for fructose 1,6-bisphosphate will undergo splitting to form 3-carbon compounds. 2 3-carbon compounds. One is PGAL, and second is DHAP, dihydroxyacetone phosphate. Is that clear? Phosphoglyceraldehyde. Now, these two compounds formed here, they are interconvertible. They can change from one to another form. Now, let's see what happens for PGL. The same thing will happen for DHAP. Why? Because DHAP can change in this. Is that clear? So, this 3-PGA will change into another 3-carbon compound known as 1,3-BPG. This 1,3-BPG. Are you getting that? Now from 1,3-BPG, now it will become 3-PG. Please see here. This 1,3-BPG has two molecules of phosphate and 3-PG has only one phosphate molecule. It means one phosphate has been taken out and formed into and changed into ATP. Yes or no? So one phosphate was taken out of 1,3-PPG, added into ADP and resulted in the formation of ATP. Yes or no? Similarly, now 3-PG will change into 2-PG, they are isomers. Now 2-PG will change into PEP by the addition of water, and then PEP will change into pyruvate. So this is phosphoenol pyruvate and this is pyruvate. So we are going to take out one phosphate from here and again become ATP. Yes or no? Please tell me that these points are clear. Yes or no? See, during this, and now same thing happens for DHAP. So can we say everything is happening twice? So whatever was happening for 3-PGAL, same thing is happening for DHAP. So we can just multiply everything with 2. So how many ATPs have been produced? How many ATPs has been produced? 4 ATP produced at the expense of 2 ATP. So net gain, direct net gain of ATP, 2 ATP. Can we say that? So these are the energy yielding processes. Are these clear? Now, as I told you, that one glucose molecule which underwent glycolysis, it formed 2-pyruvate, right? So one 6-carbon compound broken down to 2 3-carbon compounds. 2 3C compounds, 3-carbon compounds. Is that clear? Now this pyruvate, either it will undergo anaerobic respiration and form lactic acid and alcohol, or alcohol, but then that would happen in cytoplasm. But if the fate is to go through aerobic process, now in that case, this pyruvate will happen to enter into the mitochondrial matrix. Do you see it? It will enter into the mitochondrial matrix. Yes or no? Now the first reaction which will take place in the mitochondrial matrix would change pyruvate into acetyl coenzyme A. Yeah, right? So these are the equations. So see, pyruvate is a 3-carbon compound. It loses one CO2 and forms 2 acetyl coenzyme A. So 1 carbon dioxide is produced here. And during the process, NAD positive changes into NADH plus H positive. Now since there are 2 pyruvates, it will form 2 acetyl coenzyme A and 2 carbon dioxide will be released, and 2 NAD positive will change into 2 NADH, and the enzyme used during this conversion is known as pyruvate dehydrogenase. Is that point clear? Now, it is acetyl coenzyme A which can enter into a Krebs cycle. Acetyl coenzyme A will happen to enter into the Krebs cycle. Please tell me that these points are clear to all. Now see, acetyl coenzyme A will be accepted by oxaloacetic acid, which is a 4-carbon compound. Is that clear? Now, oxaloacetic acid is a 4-carbon compound. We have seen that in C4 cycle also. Acetyl coenzyme A is a 2-carbon and oxaloacetic is a 4-carbon compound. 2 plus 4 becomes 6. So here, the 6-carbon compound, that is citrate, is produced. The first compound of Krebs cycle is citrate, that is why, or citric acid, that is why we call it a citric acid cycle also. This is another name for Krebs cycle. So generally the name of the cycles is on the base of the first stable compound form. Now citrate will become isocitrate. Isocitrate is again a 6-carbon compound. Yes. Now this 6-carbon compound will lose CO2. Watch it carefully. This 6-carbon compound will lose CO2, and how many carbon dioxides are left behind? 5 carbon. It will form a 5-carbon compound known as alpha ketoglutaric acid or alpha ketoglutarate. Is this point clear? Is this point clear? Yes or no? Okay. Now, the 5-carbon compound is again going to lose another CO2 molecule, to become a 4-carbon compound, which is succinyl coenzyme A, right? And during the process, NADH is produced. Now, watch carefully. NADH was produced when isocitrate changed into ketoglutaric acid. Second NADH is produced now when 5C changes into a 4C compound, succinyl coenzyme A. Now, when succinyl coenzyme A changes into succinate by losing the coenzyme A, now here a GTP molecule, you can consider it equivalent to ATP. They are not. There is a mechanism to convert GTP into ATP, but let's make it easy for a while. So GTP is produced, one molecule of GTP. Is that clear? Now, It changes into succinate, which is a 4-carbon compound. Now, this 4-carbon compound will change into another 4-carbon compound known as fumarate. And during this conversion, first FADH2 will be formed and the only FADH will be produced. Yes or no? Please tell me. Is this point clear? Then fumarate, on getting water, getting hydrated, it changes into malate. And malate is also a 4-carbon compound. Then malate undergoes oxidation and forms another or third molecule of NADH and becomes oxaloacetic acid again. So again, oxaloacetic acid has been regenerated. Please tell me that these points are clear to you all. Please tell me these points are clear to you all. Yes or no? Right? So during the entire process, during the entire process, how many NADH have been produced? 3. How many FADH have been produced? 1. How many GTP have been produced? 1. 1 GTP. Now this is for every acetyl coenzyme A entering the Krebs cycle. But how many acetyl coenzyme A do we have? 2. So in all, there are 2 Krebs cycles which will be running after 1 glucose molecule. So for 1 glucose molecule, you need to run 2 Krebs cycles to break it down completely. Are these points clear? And now see how many CO2 have been released during the Krebs cycle? 2. And 1 was released during the link reaction. So overall, link reaction plus Krebs cycle, how many CO2 have been released? 3 CO2. So this is for 1 glucose molecule. Sorry, this is for 1 pyruvate. How many pyruvate do we have? 2. So how many CO2 will be produced? So for 1 pyruvate, if 3 CO2 are released, so for 2 pyruvate, 6 CO2 will be released. So this is the reaction that we have been writing. 1 glucose will break down to release 1 molecule of glucose, right? And 6 molecules of CO2 will be produced. Yes or no? So this is how these 6 molecules of CO2 are produced. Are we getting it right? Are we getting it right? Now see, you get a question, this is a previous year question, that in which of the following reactions oxidation has taken place? In which of the following reactions of Krebs cycle oxidation has taken place? See, every reaction where NADH and FADH is being produced is an oxidation reaction, So 4 steps where oxidation is taking place. Please let me know that these points are clear. Is that clear? Okay, so this is how you can calculate. And now let's see it quickly. See this says, so the FADH and the NADH which has been produced, now these are going to enter into the electron transport system. They are going to enter another into another process which is called ETS. Now this occurs in the inner mitochondrial membrane. So let's say this is the inner mitochondrial membrane. This is the inner mitochondrial membrane, all of it. This is the inner mitochondrial membrane. Is that clear? And this one being the outer mitochondrial membrane. I have just made the inner mitochondrial membrane broader to make you see what is happening inside the inner mitochondrial membrane. Clear? And we also know that the space between the outer and the inner mitochondrial membrane is known as a perimitochondrial space. So just like I was mentioning that you must note the location of molecules in photosynthesis, the same thing you have to do it here. You must note where each and every character of the drama is present. Is that clear? Now see, let's focus on some complexes. Now ETS uses 5 complexes. There are 5 complexes present in the inner mitochondrial membrane. Right? Is that clear? Now the 5 complexes. The first and the foremost question that you get in exam, you should know the name of the complex. So see, this is complex number 1. Complex 1 is NADH dehydrogenase. Complex number 2 is succinic dehydrogenase. Where is this present? It is present on the inner side of the membrane towards the matrix. This is inside the matrix. Why? Because the succinic dehydrogenase will be used. It is used by electron transport system as well as Krebs cycle. This is a common enzyme or a complex between ETS and Krebs cycle. Please see that. It is a common thing between ETS and Krebs cycle. That is why it is present towards, not actually in the matrix, yes, towards the matrix, but near to the inner membrane, so that both can utilize it. Are these points clear? Now, the third complex is cytochrome BC1, and the complex number 4 is cytochrome C oxidase, while complex number 5 is ATP synthase. It is the same ATP synthase which we have studied in thylakoid membrane and was helping in ATP production. Here also the same ATP synthase, just the polarity will change. We'll see how. Please tell me that these points are clear to you all. So first of all, you need to know where are they present. They are present in the inner membrane except for the succinate dehydrogenase, which is connected to the inner membrane but present in the matrix. Is that clear? The names of these complexes should be very, very clear and learned by heart, because this is a direct PYQ. Match the following complex 1 to its real name. Is that clear? Now, see, what is this complex 1 made up of? It is made up of flavin mononucleotide, FMN, and it has iron sulfide, FMN, and to FES. First, just see what they are made up of. Then we have the second complex, which is made up of FAD, flavin adenine dinucleotide, right? So FAD, and it also has FES. Talking about complex number 3, see, it has cytochrome B, it has ferrous sulfide, and cytochrome C1. That is why the complete name is BC1. So it has B, it has FES and C1. Clear? And then we have a small molecule protein, cytochrome C, present on the outer side of inner membrane. This is cytochrome C. It is a small molecule here. And then there is complex number 4. We call it cytochrome C oxidase, which is copper containing. This is copper containing. The only copper containing complex here is cytochrome C oxidase. See, copper ion, cytochrome A1, cytochrome A3, then again copper ion. Is this point clear? Now, if you have understood all these things, now let's see, why do we call it electron transport system? We call it electron transport system because electrons are going to be passed from one place to the another. So I hope we all have played that game, when we pass pillow from one person to another and the final person who gets the pillow has to do an activity, right? So this is how they are also going to pass on these electrons. So first of all, see, we have this molecule NADH. This NADH is going to lose one electron. See, NADH was produced in the matrix. FAD was produced in the matrix. So what is the final destination? What is the final destiny of these two molecules? Now see this. First, we'll talk about the destiny of NADH. See carefully. This NADH loses one electron and becomes NAD positive and H positive. Is that clear? Now, this electron lost is picked up by FMN. Is picked up by FMN. Yes or no? Now, FMN, wait a second, guys. This FMN, just a second, guys, just a second. This FMN is going to pass that electron to FES. It is going to pass it on to the FES. It is going to pass it on to FES. Is that clear? Now, from FES, this electron will be passed on to, wait, let me use the highlighter. Yes, guys. So the electron produced here, this is passed on to FMN. From FMN, it goes to FES. From FES, it goes to ubiquinone. Is that clear? Now, ubiquinone is present within the inner mitochondrial membrane. Now, this ubiquinone will pass it on further to complex number 3. Do you see this? From complex number 1, the electron directly goes to complex number 3, cytochrome BC1, through ubiquinone. Is that clear? Then from complex number 3, see, to complex number 3, cytochrome B, then FES, then C1. From complex number 3, it goes to cytochrome C. And from cytochrome C, this electron is handed over to cytochrome C oxidase, that is a complex number 4. Copper ion to A1, then A3, then another copper ion. And now finally, from the copper ion, this electron will be accepted by oxygen. Will be accepted by the oxygen. See, we are talking about aerobic respiration, but by far oxygen has not come into the scene. So during the last step, this last step of electron acceptance is done by oxygen. So oxygen is the electron acceptor. Oxygen is like the showstopper. You know, the whole show is named after the showstopper. But when does the showstopper come? At the end of the program, right? So that is what our oxygen is. It is a showstopper. So during the whole process, which is named after oxygen, okay, oxygen is because of oxygen we call it aerobic process, but this is the step, the terminal process. It is the last step. It is the terminal step where oxygen comes in. And what is the function? It is the last electron acceptor. So from complex 4, that is cytochrome C oxidase, electron is given to oxygen, and oxygen will take this electron and form water. Is that clear? Is that clear? Now, similarly, this FAD, just 2 more points, 2 more minutes. This FAD will also, FADH2 produced during the Krebs cycle will also lose an electron. Now again, this electron will be accepted by, FAD. From FAD goes to FES. From FES it goes to ubiquinone. From ubiquinone it goes to this complex number 3. From complex number 3 it goes to cytochrome C. From cytochrome C again goes to complex number 4. And from 4 again, who takes it over? It is oxygen which will hold it, accept it. And it will accept electron to form again water molecule. Are these points clear to you all? Now please see, watch carefully that during the oxidation of FADH2, which all complexes were participating? It was complex number 1, complex number 3 and complex number 4. Yes, these are the 3 complexes which are participating. Now, during FADH2, which all complexes were participating? Complex number 2, complex number 3, and again complex number 4. So 3 and 4 will participate anyhow in both the processes, in the oxidation of both NADH as well as FADH2. But complex 1 is only for oxidation of NADH, and complex 2 will only participate in the oxidation of FADH2 process. Now, you might ask, where is ATP? Now see, guys, please understand that nothing happens for free in this life. Nothing is free. No free lunches at all. So if the complex 1, see, if the complex 1 is letting the electron to pass through, or they are transporting electrons from one to the another complex, another molecules, they are doing it at the expense of something. So while electron is being transported from complex 1, there are 4 H positive ions which will be moving out. Is that clear? Similarly, when electrons are being transported by complex 3, there are 4 H positive ions which will be pumped out. Similarly, for complex number 4, H positive ions will be pumped out, but not 4. 2 H positive will move out of complex number 4 at the expense of transporting the electrons. Are you getting that? Now, again, that same chemiosmotic hypothesis comes in. Same comes in here. So, see, for oxidation of NADH, we know who all participated. Complex 1. How many H positives protons will be pumped out? 4 protons will be pumped out here. How many are pumped out by complex 3? 4 here pumped out here. And how many will be pumped out by 4? 2. So total H positive ions or protons pumped out during the oxidation of NADH are 10. So for one NADH oxidation, 10 H positive are pumped out. Is that clear? Now similarly, let's note it for FADH2. So when FADH2 is being oxidized, complex 2 was participating, but complex 2 does not transport any protons. So nothing here. While complex 3, we know 4 H positive ions, and complex 4 will transport 2 H positive ions. So for oxidation of one FADH2 molecule, we understand 6 H positive ions are pumped out. Where are they getting pumped out? In the perimitochondrial space. Can we say that perimitochondrial space will have high H positive concentration? This will have high H positive concentration. Can we say that? Yes. And in the lumen, because they are getting pumped from the matrix, so matrix will have low H positive concentration. Can we say that low H positive concentration in the matrix? Yes or no? Please tell me. Can we say that low H positive concentration here? Everybody. Is this clear? Right. Now we understand there is complex 5, which is ATP synthase, and it knows how to pump out the H positive ions from higher concentration to the electron to the low concentration that is in the matrix. Now see, the polarity of ATP synthase is different now. It again consists of two components, that is F naught. This is F naught. We'll not call it CF naught because it is not a component of, it is not a part of chloroplast anymore. So C represented chloroplast. We'll just call it F naught, and the other part is F1. And F naught is always transmembrane bound protein, so it will act as a channel to transfer H positive ions from here into the lumen back. Are these points clear? It will transport. Yes or no? Clear? So while this transportation is taking place, we understand for every 4 H positive ions, for every, see, NADH oxidation resulted in transport of 10 protons, right? And FADH resulted in FADH2 oxidation resulted in transport of how much? 6 H positive ions. For every 4 H positive ions pumped out, 1 ATP is produced. So can we say this is equivalent to 2.5 ATP and this is equivalent to 1.5 ATP? Can we just round it off to 3 ATP? And can we round it off to 2 ATP? So can we say that one FADH2 molecule is equivalent to 2 ATP and one NADH is equivalent to 3 ATP? Yes or no? Please tell me if these points are clear to you. Please tell me that these points are clear to you. And in the end, guys, we have this interrelationship among the metabolic pathways where we see no matter what the energy substrate is, but ultimately they all come down or they join that common pathway, some directly or indirectly. So when carbohydrates break down, we know they form simple sugars and gradually form pyruvic acid, and pyruvic acid will form acetyl coenzyme A and enter the Krebs cycle. Yes? But when proteins break down, they will first break down into amino acids, and ultimately amino acids will follow some metabolic pathway and join this pathway again and become pyruvic acid. So amino acids are going to join the pathway by forming pyruvic acid. While if we talk about fats, we know fats are made up of two things. We have discussed that yesterday: fatty acids and glycerol. So fatty acids will break down to form acetyl coenzyme A. This was 2023 question. While glycerol will break down to form DHAP, and we know DHAP is an important compound of glycolysis. So ultimately, glycerol is going to join the glycolytic pathway through DHAP. Is this clear? So you see, can you note down which is one molecule which is common in all the energy substrate? It happens to be acetyl coenzyme A. Whether it is fats or carbohydrates or proteins, acetyl coenzyme is a common intermediate during the breakdown of carbohydrate, fat and proteins. Are these points clear? And you must know that body loves carbohydrates, right? So it breaks down the carbohydrates in the first place, then it goes on to breaking down fat and finally proteins. Is this clear? Is this clear to all? And last not the least, we have respiratory quotient, which is the volume of CO2 evolved upon volume of oxygen consumed. And this RQ, respiratory quotient, it depends upon the substrate used. For example, for carbohydrates, the value is always 1. For fats, it is less than 1, around 0.7. This is for tripalmitic acid. Then for proteins, it is around 1 again, but less than that, but near to 1, that is 0.9, and for organic acid it's always greater than 1. So for example, you are taking lactic acid or oxalic acid or citric acid, any organic acid, the value of respiratory quotient would be more than 1. Are these points clear to all? So guys, with this, we complete our human physiology. Now, please don't forget to kickstart your NEET preparation by registering for the free NEET mock test series, okay, which will be held from Sunday onwards, from 26th onwards, every day. See, it happened last Sunday, but now on from this Sunday onwards, it is going to it is going to take place every day from 2 to 5 p.m. So make sure that you attend those, you take those mock tests. Even NTA is notifying the students, guys, that please appear in the mock test before coming for the final exam. Go for the NEET drill. So make sure, listen to NTA, listen to everyone, and take those tests from this at the same time. At the same time, that is 2 to 5 p.m. Get your body ready, get your mind ready. Is that clear? For the same time and same condition. And of course, it has other benefits also. I'll see you tomorrow. I'll see you tomorrow with the next unit, and we will be discussing the plant physiology is done. Now we are left with human physiology, and we'll be completing it tomorrow. Thank you so much. Have a lovely night. Keep revising. I am done with NCERT reading. Revise the chapters again after attending the session. Thank you so much, and have a good night.